Today is Wednesday, November 17th. We are on to our second lecture of the nervous system. We got a lot of nervous anatomy out of the way in the last class, so we get to focus on the function of our nervous tissue now. And today we're going to produce those potentials we've been talking about for so long. In lab today, we will start, we did our introduction to the brain anatomy, and we'll talk more about those cephalons and the regions and the areas and the functions that I threatened you with last class. Handful of assignments coming up. Next week, we do have class. On Monday, your unit 12 is due. On Wednesday, I know it's the day before Thanksgiving and everybody wants to be prepping for the holiday, but unfortunately it is one of the days that we meet and this section is packed. And we, again, I didn't do this on purpose so that this would be punitive. This is just the way that the schedule fell out. We do, and so that's why I'm making you aware of it ahead of time. Again, I usually surprise you with the daily quizzes, but I'm telling you right now, there's going to be a 5-point daily quiz on Wednesday, the 14th and the 24th. And more importantly than those 5 points is we are going to be discussing the cranial nerves, which, as I mentioned, depending on how far behind we get in this section, could be as much as 25% of your lab exam. And anything that important is if it's 25% of your lab exam, do you think it might find its way onto your lecture exam as well?
Yeah.
Yeah, absolutely. So it's going to be some important material we're going to be covering on that. Then the following week, we have 2 big assignments due. Your PhysioX exercise 3, which has 9 activities. And again, at this point, you've realized the PhysioX isn't hard. It isn't challenging. It's just time consuming. So it takes a bit of time to work your way through it. And with 9 activities, it's going to take even more time to work your way through it. And lastly, well, that's on the schedule anyway. For the December 1st, we have a nervous system review. Again, one of the points that we started emphasizing in the last class is this relationship between structure, function, and location. Now, those 3 things go together. And that 15-point nervous system review, which I have already posted, but hopefully you haven't started working on yet, because we haven't talked about many of the locations yet. But as we put those locations together in the spinal cord and other parts of the body and put them together, we understand this relationship. And so the point of this exercise is to emphasize that relationship between structure, function, and location, and it will be graded for correctness. And then other than that, we have one more daily quiz coming up at some point, and then one more unit review, and then a fun reflexes lab. And that's it. That's the extent of what we have going on for this semester. So we are rapidly approaching the end. All righty, questions on any of that? All right, excellent. So we left off last class and we had talked, like I said, about the anatomy of the neurons. We talked about the 3 structural classifications, the 3 functional classifications, and things along those lines. And we also talked about their characteristics, how like muscle tissue, they're irritable. They're capable of producing an electrical signal. And we call that electrical signal a potential. Now, what we haven't been emphasizing up to this period of time is that there's actually more than one type of potential. There is the one we've been talking about since practically the beginning of this class, the action potential, that big all or nothing signal. All right, it's like the light switch. I have that light switch, and if I just push on the light switch just with the tiniest bit, if I just touch my finger to the light switch, does anything happen?
No.
No. But if I generate enough force with my finger, it flips the switch and the light comes on. However, if I flip the switch twice as hard, does the light come on twice as bright?
No.
No, trick question. Yeah, no, of course not. Right. So it is a standard big size. As we've said before, the action potential is about a 100 millivolt change. It is a big positive signal. Big positive, oops, standardized. Oops. Signal. Now, Remember, we also mentioned in the last class that these only occur along the axons. The axon has a specialized plasma membrane. And what did we call that special plasma membrane of the axon?
Axon.
There we go. Excellent. Axolemma. Perfect. Where it's able to produce that big positive signal. Right? So it's all or nothing. It's on or off. And like I said, when I flip the switch, if I hit it twice as hard, it doesn't come on twice as bright. If I hit it just gently, I don't get half the luminescence. It's just one thing, on or off. However, is every light switch exactly like that?
No.
No. I'm sure, like my dining room, your dining room has a special little knob that allows you to be able to adjust the intensity of the light, right? If me and my wife want to have a nice romantic dinner, we turn the light up nice and bright so that we can see each other and enjoy each other's company and enjoy the food, right? When our kids are sitting down at the table with us, we turn it down nice and dim so we don't have to look at them and the mess that they make, right? We have that ability to adjust, to be able to modify it. And that type of electrical signal occurs in our neuron as well. These are graded potentials. These are small signals. The average graded potential is around a 2 or -2 millivolt change. Very, very different from the 100 millivolts that we see with an action potential. And notice I said average. Notice I said variable. It can be 10, it could be 15, it could be 1, it could be half. And notice something else. I said it could be a +2 or a -2. Unlike the action potential, this can be a positive or a negative signal. So not only can it vary in intensity, but it actually has the ability to be negative as well as the ability to be positive. Now, these are small, tiny waves. And if I sit at the end, as I've said before, right, being a community college instructor, I have massive scratch. So I have a huge Olympic-sized pool in my backyard. And if I just sit at one end of it and I just tap the water and produce tiny little waves, are they going to travel very far? They're going to reach it all the way to the other end of my massive pool in the backyard? No, of course not. Where if as I take that car and throw that car into the pool, I produce a big wave and it easily makes it to the other side. Right? The other thing about these action potentials is they can travel long distances. Like the meter it takes to get from the small of your back to your big toe. However, these graded potentials, being so small, only travel a short distance. But that's okay, because these are produced along the dendrites and the cell bodies. And any idea what the goal of these graded potentials is going to be? We're going to produce a little wave. Where do you think that little wave needs to get? Any idea where that little wave may need to get to have an effect? True, but they're not going to help us get to another neuron, but an action potential would help us to get to another neuron. You've absolutely got the right idea, Sarah. Our goal is to communicate with another neuron. So we need to use these graded potentials to make an action potential. And where would we trigger an action potential at?
Trigger zone? Or
There you go, exactly. The trigger zone, which is in the axon hillock. Absolutely excellent. Our goal is to get to that special region, that trigger zone, where we're able to produce an action potential. And if we can use these graded potentials to stimulate that trigger zone enough, boom, we get our action potential, and our action potential spreads down the neuron. All right, so notice there are some big differences between these two types of potentials. But a potential is still, if we think of it in electrical terms, it is still a current. Oops. And a current is basically the movement of a charged particle. You have a current running along that wire from the light switch to the ceiling light. And when you flip that switch, an electron, a charged particle, moves along the wire. And as it moves along the wire, that current is what turns on the light. And so it's the same thing here. We want to move charged particles. Of course, here in a neuron, it's not an electron that's moving. What is the charged particle that we're moving? Come on, what are the things that we've been talking about since practically the beginning of this class that have charges to them?
Ions.
Yeah, ions, exactly. Cations and anions. We move that charged particle, in this case an ion, and we move it across the plasma membrane, either into or out of the cell. And if we do that, we produce our current. We produce our potential. Of course, to move an ion either into or out of the cell, we need two things. And these are two things that we've already talked about before. A neuron needs to have a resting membrane potential. It has to have a difference in its charge inside or out of the cell. And it has to have ion channels. So let's do this on the whiteboard. Is there an unequal distribution of ions inside and outside of a cell?
Yeah.
Absolutely. Right. Where is there more sodium?
Outside.
Outside. Excellent. What's another ion that is unequally distributed across the cell?
Calcium.
Calcium. Where is there more calcium?
Outside.
Outside. Excellent. Give me another one.
Chlorine. Outside.
Excellent. Chloride. That's our first anion. And what's the other one we've talked a lot about?
Potassium.
Where is
Inside.
there more of that?
Inside.
Inside the cell. Absolutely. We compare that to very little sodium inside, very little calcium inside, very little chloride inside, and very little potassium. So as we know, there are driving forces, right? Basically what we talked about before are concentration gradients. That want to drive the cells. Because remember, one of the key things that we need to remember is that all ions want to be at equilibrium. Ions are like a 3-year-old. What does a 3-year-old care about?
Themselves.
Yeah, all they do is they care about themselves. Absolutely. They don't care about the cell. Sodium doesn't care about potassium or calcium or chloride or anything else. All sodium cares about is itself. And so as we've talked about, there is a concentration gradient where cells want to be at equilibrium. Ions, pardon me. So based on that concentration gradient, or what we can call our chemical force, which way does potassium want to move?
Outside as well.
Out of the cell. Which way does sodium want to move?
Inside.
Calcium wants to move.
Inside.
And chloride wants to move.
Inside.
There you go. All right. But remember, we also talked about how with this unequal distribution of ions, there is also an unequal distribution of charge inside and outside the cell. Where is it more positive, inside the cell or outside the cell?
Outside.
Exactly. So here outside the cell is more positive, and it's inside the cell. And we were actually able to give a number to that, right? When the cell is happy, when the cell is at rest, it has a resting membrane potential of what?
-70 millivolts.
-70 millivolts. Perfect. Now, ions are charged particles. Like magnets, positives don't like other positives, negatives don't like other negatives, but a positive and a negative, they will attract. So there is also what we can call an electrical force that is going to drive these ions as well. Potassium being positive, its electrical force, does it want it to come in or does it want it to come out? In. Chloride, does it want to go in or does it want to go out? Out. Excellent. Calcium, in or out?
In.
Sodium, in or out?
In.
Yeah. Notice both sodium and calcium really, really, really want to come into the cell. Both their chemical and their electrical forces want them to come in the cell. Now remember, calcium makes cells do wonky things. So do we necessarily want calcium coming in all the time? Or staying around inside the cell? No, so remember we have pumps and other things that try to get that calcium out. But remember, we also talked about with those co-transporters, because sodium really wants to come inside, remember we were able to use this to do some work. So we're able to take advantage of some of these things so that the cell can do some work. So that's the real key here. There are driving forces on our ions. Ions want to be at equilibrium. However, at the same time, I want to be in Fort Knox, right? Because it is filled with gold and you can actually, like Scrooge McDuck, dive off the diving board and swim around in all the gold coins that they have there. So why am I not at Fort Knox doing that right now? I mean, aside from the fact that it's in Kentucky. Yeah, they're not going to let me in the door. So you can have all the driving force you want, but if the ions can't move into or out of the cell, they're not going to be able to do work. So the other thing we need, which again, we have talked about in this class, is we have to have channels. And there are two types of channels that we can find in a cell. The first are what are called leak channels. Guess what the state of a leak channel is?
Is it open?
Yeah, it's always open, absolutely. Right? If you have a leak in your ceiling, is that only open some of the times?
I wish.
Exactly. No. So a leak channel is exactly what it sounds like. It's basically a hole in the cell that an ion can pass through. And the cell has a lot of them. Remember, equilibrium is not where there's no movement. Equilibrium, remember, is no net movement. Even when a cell is at rest, there's a massive amount of movement of ions that are taking place. For instance, there are sodium leak channels. And from what we know, what is sodium constantly doing in a sodium leak channel?
It's leaving.
Does sodium want to leave the cell or does sodium want to enter the cell?
Enter.
Enter the cell, absolutely. What about a potassium leak channel?
Outside.
Yeah. Potassium is constantly going to be leaking. And while we haven't mentioned that per se as much, if you also remember, one of the things that we said when a cell is at rest, that sodium potassium ATPase is responsible for uses about 25% of the ATP of a cell when it's at rest. And remind me again what that sodium potassium ATPase does.
It's out 3 sodiums and 2 calciums, or 2.
Yeah. Okay. 3 sodiums and 2
Oh, sorry,
potassiums.
yeah.
That is correct. Which direction do they move those sodiums? There we go. Excellent. Perfect. Yeah. So it kicks 3 sodiums out. And it brings 2 potassiums in. So that sodium basically leaks in and then is kicked right back out. That potassium leaves and then is basically pumped right back in. However, notice 3 positive things leave and only 2 positive things come in. So this also helps to maintain the resting membrane potential, helps to keep the cell negative. Right? Remember we said the resting membrane potential is about negative 70 millivolts. Oops. It turns out that almost 15 of those millivolts are just from the sodium potassium ATPase. We actually know this because scientists have actually used a chemical to block the sodium potassium ATPase, and suddenly the resting membrane potential goes up to like negative 55 millivolts. Again, you don't have to know that number, but just the point is these leak channels, there's constantly ions moving in and out of the cell, either being pumped out or in through these leak channels. However, if it's a leak channel, is it something we can control?
No.
No. And if we can't control it, it's not going to help us to do work. Luckily, as we have learned, there is a second type.
Could you go back off of it, please?
Yeah, hold on.
Okay.
So the point is these leak channels are always open. And since they're always open, they cannot be changed. And if they can't be changed, they can't be used to do work. All right.
Thank you.
Yeah. So luckily for us, there is a second type of channel. And these, as we know, are gated channels. And it turns out there are 3 types of gated channels. And believe it or not, we have learned all 3 types. So let's talk about them. Here's my plasma membrane. And there are 3 types of channels. Any idea what those 3 types of channels might be? Give me one of them. Voltage gated, excellent. Voltage gated is a fun one we have talked about. Absolutely. It is made up of proteins in the plasma membrane. And what's special about these voltage gated channels, and I'll draw it this way. Like all channels, it is made up of proteins. And in particular, this has two big proteins that have a tremendous amount of positive charge on them. So when this cell reaches a special critical point, and what would that special critical point be? There we go, threshold. And anybody remember what we said the value of threshold typically was?
-60 millivolts.
There you go. So when we get it a 10% positive, 10 millivolts more positive, when it reaches threshold, those proteins, or what we call the activation gate, swings open. And so basically what happens is this is going to, when we reach threshold, these gates swing open. And when they swing open, then our ions are able to flow. There you go. Voltage gated channels are something we've talked about before. Threshold is something we've talked about before. However, there is one more thing about these voltage gated channels we need to mention. Voltage gated channels actually have 3 states. The first is closed. The second is open. But some voltage gated channels have 2 additional proteins. One is a long linear protein, and one is a big globular protein. This is known as the inactivation gate. And because of the shape of its proteins, it also gets a very, very technical scientific name. It is also known as the ball and chain. What happens is that when this channel is open, when these gates swing open, this ball can actually come and wedge itself into the channel. When this, and again, I haven't drawn my scale correctly here, but I'll cheat. When this ball is wedged into the channel like that, even if our gates are open, are ions going to be able to flow into this channel? No. And so we call this state inactive. So it has 3 states. It can be open, it can be closed, and it can be inactive. And there's one other problem when it's inactive. When this is inactive, it cannot open again until the channel closes. So basically, what has to happen is that when the gates swing down, they pop the ball out. And then the channel can be used again. When it's in that inactive state, basically the channel can't be used. So this is another layer we're adding to that voltage gated channel we learned about. All right, questions on that? I know my pictures aren't the best. I have better pictures, and your textbooks have some great pictures. I'll show you in just a minute. What's another example of a gated channel we have already learned about in this class?
Chemically gated.
Excellent. With a chemically gated channel, we have a protein that has a binding site for a chemical. And when no chemical is bound to it, it is closed. However, when the right chemical, like for instance, acetylcholine, comes in and binds to it, then that causes the channel to open. And when the channel is open, ions can flow in and out. So again, with a chemically gated channel, a chemical, or what your book also refers to as a ligand, remember, which is basically just a chemical structure, molecular structure. Where that chemical or that ligand binds the channel and opens it. Excellent. And we've seen examples of those, like the chemically gated cation channel that we've talked about. But believe it or not, we have also already talked about the third type of channel. The third type of channel is a mechanically gated channel. I know that isn't a word we've used already in this class, but believe it or not, we did talk about it. If you remember, oops, didn't want that. If you remember way back when we were talking about those pressure receptors in our skin, and what were the name of those pressure receptors of our skin again?
The tactile corpuscles?
No, the ones for the pressure, right? That was the fine discrimination. What was the one that when you squeezed kind of looked like an onion, remember?
Lamellated corpuscle?
There you go, lamellated or Pacinian corpuscles. Those have what are basically mechanically or stretch gated channels. This is a channel that when the cell is at rest, basically sit in a closed state. However, when you put a force on the plasma membrane, when you stretch or push on the plasma membrane, that causes the gate to swing open. And when the gate swings open, then the ions can move in and out. And when you remove the pressure, what happens to those two channels? Those two proteins, they go back together. So basically, when you put pressure on the plasma membrane, it pulls them apart. When you remove that pressure, it goes back together. So even though we didn't use the term mechanically gated channels, we talked about how when you stretch those layers of the onion, we perceived that pressure. And again, I fully appreciate my drawings are horrible here. So let's look at the pretty pictures from your textbook. Again, as we mentioned, our cell is polarized, unequal distribution of ions. That sodium, potassium, ATPase helps to maintain the membrane potential. And as we talked about, potassium leaks out and is pumped back in. Sodium leaks in and is pumped back out. And it keeps the cell at a negative 70 millivolts. But again, those leak channels are important, but they can't be changed, so they can't do work. So what we need is a way to move ions into and out. We have those leak non-gated channels that are always open, and we have some gated channels. Here are some simple illustrations of those three types: voltage, chemically, and mechanically. Here is a much better view of a chemically gated channel. Again, here's our protein in the plasma membrane. Our ion can't pass through it, but when the right chemical, and notice they used acetylcholine, when it binds, it opens, and the ion can flow through. Here is a very simple illustration of a voltage gated channel. And notice the key to this voltage gated channel is it has two gates. It has the activation gate and the inactivation gate. At rest, the activation gate is closed. And when this cell reaches threshold, that causes the activation gate to open. Now all the channels are open, all the gates are open, I should say. The channel is open and our ions can flow. But remember, we mentioned how there is this ball and chain. And this ball and chain can actually wedge itself into the channel. And when that occurs, it is now inactive. Even though the gate is open, ions cannot flow. And remember, the other big key to this is the channel must close before it can be opened again. You cannot go from an inactive state to an open state. It has to close first. This simple illustration doesn't do a good job of showing that, but the picture from your textbook does a great job. I'll show you that in just a minute. But before we do that, here's a better view of that mechanically gated. Again, here at rest, the channel is closed. When we put pressure on it, it opens. That pressure is removed, it closes again. All right, and then here, as promised, is that much better picture of a voltage gated sodium channel in this case. Again, notice here is our activation gate. Here is our inactivation gate, that ball and chain. When the inside of the cell gets positive, it causes that highly positive activation gate to shift its position. And when it shifts its position, now the channel is open. And now our ions can flow. But this ball is actually just bouncing around randomly. Some molecular neuroanatomists have actually gone in and mechanically changed the length of this chain, made it longer and made it shorter. And what they found is when they made it shorter, it inactivated faster. When it was longer, because it was swinging around randomly, it was longer before it happened to wedge itself in there. But eventually, what's going to happen is that that ball is going to wedge itself in there. And when that ball wedges itself in there, ions can no longer flow. And the only way that the cell can get this ball out is to repolarize. When it repolarizes below threshold, then what happens is that our gates swing down. And when our gates swing down again, then they pop the ball out. So once the cell is inactive, once the channel is inactive, the only way it can be used again is if the channel closes first. And as we'll see, that is a very important rule. We have to close the channel before it can be used again. All right. Questions on this? All right, excellent. So there's one last piece we need to add to this puzzle. As I have already mentioned, ions are like 3-year-olds. All they care about is themselves. So when you open an ion channel, all that ion channel cares about is reaching its equilibrium, where it's going to be happy. So what that means is we need, if we know what that equilibrium is, we know how ions are going to flow. So let's learn some numbers. As you've seen in this class, I'm not huge on numbers because numbers are usually things that can be looked up and they aren't vitally important, but there have been some numbers that have been important. We need to know a sodium potassium ATPase moves 3 sodium out of the cell and 2 potassium into the cell. We also, talking about things we've talked about right now, know 2 numbers that are of importance to the cell. Those 2 numbers that are important to the cell are the resting membrane potential and what was the resting membrane potential again?
-70 millivolts.
Excellent. And another important number we've learned is threshold. And we can be more specific now. Threshold for opening our voltage gated channels. And what was the threshold for opening our voltage gated channels?
-60 millivolts.
So now we can add 2 more important numbers. We can add the equilibrium potential of sodium. And it turns out the equilibrium potential of sodium is positive 66 millivolts. So here we are with our cell happily chugging along at -70 millivolts. And again, if we leave it alone and provide it with the oxygen and the nutrients that we need, it will stay at -70 forever. But if we open a sodium channel, sodium moves to reach equilibrium. Now, notice to reach equilibrium, sodium wants to make the cell more positive. So which way does sodium move to make the cell more positive? What's a positive ion? Does it make it? Yeah, by going inside. So, and it does that by moving inside the cell. So when it moves inside the cell, it makes the cell more positive. And didn't we have a fancy word for making a cell more positive?
Polarizing?
Yeah, depolarizing. Or depolarizes, let's say that. Both are correct. Depolarizes the cell. Now, do you think it's going to ever get the cell to positive 66 millivolts?
No.
No, the cell probably wouldn't be that happy there. But if we had a cell that had 1 million sodium channels and we opened all of those 1 million gated sodium channels all at once, the cell would get to positive 66 millivolts. All right. So we already knew that. We already knew when you opened a sodium channel, sodium rushed in the cell and made the cell more positive. Now we know why. Because it's trying to reach its equilibrium potential. And we need to know one more. We need to know the equilibrium potential of potassium. And it turns out that's negative 90 millivolts. So those are two more numbers you need to know. So once again, the cell is happily chugging along at rest, minding its own business. And then we open some some potassium channels, and potassium wants to reach its equilibrium potential. It wants to make the cell negative 90. So to make the cell negative 90, which way does potassium have to move?
So down.
Well, how does potassium move to make the cell more negative?
Oh,
Inside the cell.
Yeah, potassium leaves the cell. Remember, when a positive ion leaves the cell, it makes the cell more negative. If I have $100 and I spend $10, $10 leaves my pocket, I am less rich than I was before. I have less money than I have before. So a positive ion leaving the cell makes the cell more negative. And so potassium moves, leaves the cell, makes the cell more negative. Now, this is a term we haven't talked about. Does anybody know what the fancy term we use for making a cell more negative?
Hyperpolarization.
Yeah. Hyperpolarize the cell, which means to make it more negative than resting. Exactly. So these are two more numbers we need to know.
Sorry.
What? You're muted again.
Can you please go back so I can screenshot it? The chart.
Get in there. Hold on.
Thank you. Okay, thank you. You're good.
So four voltages we need to know. Negative 70, which is our resting, negative 60, which is our threshold, positive 66, which is the equilibrium potential of sodium, and negative 90, which is the equilibrium potential of potassium. If we know those four numbers, then we can completely understand both our graded and our action potentials. And so, like I said, when we open those channels and that charged sodium either leaves the cell, enters the cell, or that charged potassium leaves the cell, we have the movement of a charged particle, which, as we mentioned, is a current. And here in our neuron, the fancy term we give for a current is a potential. Now, we pretty much literally just did this, but let's go back to that graph we were looking at before and now talk about graded potentials. Again, remember, graded potentials are small variable electrical signals They can be positive or negative. And they are primarily produced on the dendrites and the cell bodies. let's go back to the idea of opening a sodium channel. We have the cell, and the cell is happily chugging along at negative 70 millivolts. And let's say I open 2 sodium channels. What would happen?
Sodium would move up a little bit.
Yeah, sodium would come in. Would a lot of sodium come in?
No, just a little.
No, so just a little bit. So we would get a small depolarization. Do you think that depolarization would last very long?
Probably not.
No, and so then the cell goes back to rest. And what was that term we used when a cell goes back to rest?
Repolarization.
Yeah. Excellent. If instead of 2, we open 6 sodium channels, what happens then?
There's a greater depolarization.
Bigger signal. And if I open 20?
Greater.
Yeah, an even bigger signal, right? So again, remember, one of the keys is this can be variable. The way we make this variable is by opening a different number of channels. We open a lot of channels, we get a bigger signal. We open a little bit of channels, we get a smaller signal. But they're still relatively small, and they still only travel a short distance, and they still only last for a brief period of time. And then very quickly they repolarize. And if instead of opening sodium channels, I open 3 potassium channels, what would happen then? Yeah, hyperpolarization, exactly. We'd get a small hyperpolarization that would then go away. And if I open 7 potassium channels, what would happen then?
A greater.
Yeah, greater hyperpolarization. And if I open 20, an even greater hyperpolarization. Notice it only stays hyperpolarized for a brief period of time, and then it goes back to rest. Do we have a term for going back to rest?
Repolarizes.
Yeah. Notice repolarization means going back to rest. It doesn't matter the direction. It doesn't matter whether it's getting more negative or whether it's getting more positive. The key to a repolarization is repolarization means going back to rest. The direction doesn't matter. All right. With me so far? All right. One more piece to add to this puzzle. Now, if we're doing work, if we're opening different types of different numbers of channels, what types of channels do we use to produce, let's say it that way, to produce graded potentials? How many different types of gated channels did we talk about?
3.
3. What were they again?
Chemically, mechanical, and
Someone help her out.
voltage. No.
Voltage gated. There you go.
Yeah.
Excellent. So which of those 3 would allow us to produce a graded potential?
Chemical and mechanical.
Okay, chemical, absolutely. If I sprinkle 2 acetylcholine on the neuron, am I going to open a lot of channels? No. But if
No.
I sprinkle 10, I'll open more. 100, I'll open more. So notice, absolutely, chemically gated channels can be used for this. I heard you say mechanical as well.
Mhm.
can we stretch a cell a little bit, or stretch a cell a little bit more, or stretch yourself a little bit more? And the more we stretch it, the more we open. But what about voltage gated? When we reach -60, do just half of the voltage gated channels open? Just a quarter of them? 3/4? 2/5? No. Notice voltage gated channels, when you reach that threshold, all of them open. Right? It's almost like a voltage gated channel is all or nothing. Haven't we heard that term before?
Yes.
Where did we hear that term, all or nothing?
The leak channel.
No, well, no, those are just all,
Oh.
right? Where did we use the term? There we go. Sarah's got it. An action potential. Absolutely. So notice, to produce a graded potential here on the cell body and the dendrites, we use the chemically and mechanically gated channels. So we're going to produce those to get our graded potentials. And remember, we said how the axonoma has a special the axon has a special plasma membrane, the axonema, that allows it to produce action potentials. What the axon has that the cell body and the dendrite doesn't is voltage gated channels. So voltage gated channels are going to be what we use for action potentials. So here's the pretty picture from the textbook. Again, graded potentials only occur on the cell body and dendrites. They vary in amplitude. They can depolarize or hyperpolarize. And remember, fun with vocabulary. Depolarize means to go above resting membrane potential. Hyperpolarize means to go below. And remember, repolarize means to go back. It doesn't matter the direction. These are those tiny little waves. Remember, the average on these are plus or minus 2 millivolts. These are tiny, tiny signals. So they can only travel a short distance, but we only need them to reach the axon hillock. If we can get them to that trigger zone on the axon hillock, then we can produce an action potential. Excellent. Questions on this? All right, spectacular. We have made graded potentials. So now what we need to do next, and we will do this after the break, is make an action potential. So let's go ahead and take our first break. It is 8:58, so let's come back at 9:13. Take a quick 15-minute break, and I will start the recording at that time. All right, any questions before we take our break? All right, see you guys in 15 minutes. So now that we have an understanding of the graded potential, we need to talk about an action potential. As I mentioned, these only occur at the axon. And now we know why, because only on the axon do we have the voltage gated channels. We also mentioned how it is a set amplitude, a set 100 millivolt change. And again, it's because all the voltage gated channels open at the same time, so we always get the same size signal every time as well. That big positive signal, and it is able to travel long distances. Now, we do have to put a little bit of an asterisk by this, because as I mentioned, in my big, hugest Olympic-sized pool that's in the backyard, right? In fact, we're planning on buying the neighbor's house and knocking it out just so we can have 2 Olympic-sized pools back to back. Even if I take a car and I throw it into the end of that and then produce this big, massive wave, by the time it gets to the end of those 2 Olympic-sized pools, Is it going to be the same size? No, it's going to shrink dramatically as it goes. And who knows, if I'm able to buy the house behind that and get 3, then it may not even make it to the other end. We need this action potential to be able to go long distances. So yes, a big signal can travel farther, but this asterisk is because axons also have a special property where they're able to self-propagate. Basically, what this means is when they produce an action potential, that action potential travels without getting smaller. It'd be like producing a wave in the pool, and that wave continues and never gets smaller as it goes. In that case, like we talked about, I could have an axon from here to Seattle, Washington, and it would be able to travel that whole distance. And when it got there, the action potential would be the same size that it is when I first produce it. So that's going to take a little bit of trickery, and we'll talk about and see how that is. But it is still a big, huge, massive signal with that ability to self-propagate. All right, so let's start with this picture, and then we'll go to our whiteboard and do it here. Notice that in our neuron at its resting state on the axon, we have voltage gated channels. And notice we have 2 types of voltage gated channels. We have 2 types, let's say it that way. And they are voltage gated sodium and voltage gated potassium channels. Now, what do you think the biggest difference between a voltage gated sodium and a voltage gated potassium channel is?
One is for potassium, one's for sodium.
Exactly, there you go. That is the obvious difference. The biggest, most obvious difference between these is clearly which ion travels through them. So of course, through the voltage gated sodium channel, sodium travels. And which way does sodium go again when we open a voltage gated sodium channel?
Inside.
Yeah, so voltage gated sodium channel, sodium enters the cell. Whereas with a voltage gated potassium channel, which way does potassium go?
Outside.
Excellent. So difference in which ion moves, difference in their direction. But there's 2 other big differences. Our voltage, so both of these, let's start here in the middle. Oops, that's not how you spell both. Both have the same threshold of -60 millivolts. So when we reach threshold, both channels start to open at the same time. However, our voltage gated sodium channels open and close very quickly. Whereas for potassium channels, they open and close very slowly. And if you look at the illustration, there's one more big difference. Our voltage gated sodium channels have an inactivation gate. They have that ball and chain. That ball and chain protein. So our voltage gated sodium channels have 3 states. They can be open, they can be closed, and they can be inactive. Whereas notice our voltage gated potassium channel has no inactivation gate. And as such, how many states does it have?
2.
Exactly. They can be open or they can be closed. So there you go. This is our starting point. Now, let's change our view slightly. Here's our axon, and pardon me, here's our cell body with its dendrites. There's its axon. And again, right now we are solely focused right here in the axon hillock. Basically what we've done is we've taken a voltometer, which basically is just a fancy machine that measures the voltage. And we are measuring it right here at our axon hillock. And if we do that, we get a graph. Actually, let's do it this way. That looks like this. Time, whoops. Versus voltage. And while we're drawing our graph, let's put our 4 important voltages that we need to know on here. What are the 4 voltages we need to know?
Starting from the top or the bottom.
It doesn't matter, just give me any of them.
Potassium is negative 90 millivolts.
Okay,
That's
I think
the
equilibrium
equilibrium
potential
potential.
of potassium is negative 90 millivolts. Someone give me another one.
The resting membrane potential is negative 70.
Excellent. Resting membrane potential, and I'm abbreviating this. Hopefully that's okay. If you don't remember what any of these mean, just ask. Excellent. What else?
Threshold's negative 60 millivolts.
Equals negative 60 millivolts. And we need one more.
And then the
Someone help her out.
or positive 66
There you go.
millivolts for sodium.
Equilibrium
Equilibrium
potential of
potential.
sodium. Excellent. All right, so again, we leave the cell alone and it happily sits at its resting membrane potential and will sit there forever. Now, if it'll happily sit here forever, then that means we need to elicit some type of change. How do we change what's happening in this location? What's the very first thing we need to do?
The graded potentials.
Yeah, exactly. The very first thing we're going to do is produce some graded potentials. Remember, a single graded potential is only about positive 2 millivolts. So is one graded potential typically going to be enough to cause an action potential?
No.
No, think about it. If we just get a positive 2 millivolt change that briefly goes away, what's going to happen?
Just gonna go.
Yeah, nothing.
Nothing
Exactly.
really.
We got nothing. If we get a positive 6 millivolt change or a positive 8 millivolt change, none of those things make a difference. What do we need to do? To see a change, what do we need to do?
A lot of sodium needs to enter.
Okay, well, that's going to cause the action potential, but what do we need to have happen here at, there we go, Sarah's got it. At our axon hillock, we need to produce a graded potential that is big enough to reach threshold. And if we are able to produce a graded potential, a wave that we produce here that spreads to the axon hillock, and if it is big enough to reach threshold, then that's when the magic happens. And what is the magic that happens when we reach threshold?
A lot of sodium.
Okay, remember, like we talked about with the muscle tissue, we always want to talk about what the immediate effect is. There we go. Mitch has got it. When we reach threshold, voltage gated channels open. Which voltage gated channels open?
So sodium voltage gated channels.
Close.
Sodium, potassium.
Yeah. All. When we reach threshold, all voltage gated channels will open. However, I understand where you were going with your statement, Ava, because as we talked about, will they all open at the same time?
No,
No. Which ones open first?
sodium.
There you go. So the fast voltage gated sodium channels open first. And again, how many do you think there are?
Ton.
Yeah, exactly. There are tons of these. Absolutely. So what happens is a massive number of voltage gated sodium channels open. And if a massive number of voltage gated sodium channels open, what happens?
We have a big positive signal. which
Yes,
is
because a
action.
massive amount of sodium enters the cell. And when a massive amount of sodium enters the cell all at once, we get a big positive signal. And what does that sound an awful lot like? What have we called a big positive signal? You're right, it is a depolarization.
Action potential.
Yeah, it's an action potential. There you go. Process is over. We've produced our action potential. We get a big, huge, massive depolarization. And the story's over. Okay, maybe not. Fix this. All right. But we had that was our first step. Our first step was indeed to produce a big positive signal, an action potential, and we've done that. However, as we learned with muscles, if we're going to move our body through space, does it typically just take one action potential to do that? No, we want to produce many. So it's not like a neuron can fire its one action potential and then take the rest of the week off. So we need to be able to reset this so that it is able to do it again. Now, remember, what is sodium trying to reach?
It's equilibrium potential.
And what was its equilibrium potential again?
Positive 66 millivolts.
It's trying to reach its equilibrium potential, but it does not get there. And it does not get there for two reasons. No, too big. It does not get there for two reasons. The first reason is, remember that pesky ball and chain. Our voltage gated sodium channels inactivate. That ball wedges itself in there. And when it inactivates, can any more sodium enter the cell? No.
No.
So no new sodium enters the cells. And when that happens, it stops getting more positive. It peaks. And that peak is one last point, one last number we need to memorize. The peak of our action potential is positive 30 millivolts. which I'm thinking you probably could have guessed, because how big of a change did we say that an action potential was?
100.
100.
So -70 to +30, how big of a change is that?
100.
There you go. So as we talked about, this is a positive 100 millivolt change. So you probably could have guessed at what the peak was, but now we see why it peaks, because it inactivates. But notice it also isn't going to stay depolarized forever. And that's because a second thing happens. That second thing that happens is, remember, when we pass threshold, our, oops, how do I erase the threshold number? There we go. When we pass threshold, our voltage gated potassium channels started to open, but remember they were slow. Well, now finally those slow voltage gated potassium channels open. And once again, there is a ton of them. And what does potassium want to do?
Reach its equilibrium potential.
And it is super far away from that. So what is potassium going to do to reach its equilibrium potential?
Leave the cell.
Yeah.
Yeah.
And is just 1 or 2 potassium going to leave the cell?
A lot.
No, tons leave the cell. And as that leaves the cell, it causes the cell to repolarize. And so just as simply, the cell is going to go back towards its resting membrane potential. Of course, is that what potassium cares about? Being at its resting membrane potential?
No.
No, it wants to be at its equilibrium potential. So not only does it go to the resting membrane potential, but it goes past it. Let's go ahead and put these lines here so we can see them. And when it goes past the resting membrane potential to try to reach its equilibrium potential, not only is it repolarizing the cell, but what else is it doing to the cell?
Hyperpolarizing.
Excellent. So not only does it cause the cell to repolarize, but it also causes the cell to hyperpolarize. All right. Questions on that? All right, excellent. Now notice something interesting happened on the way down. As the cell was repolarizing, it crossed the threshold going down. And what happens when we cross the threshold going down?
Voltage gated channels close.
Exactly. All of our voltage gated channels start to close. However, do all close at the same rate?
No.
No. Our fast voltage gated sodium channels close first. And notice when they close, remember that pops out the ball of the ball and chain. And now the voltage gated sodium channels are no longer inactive. And remember, if they're no longer inactive, that means they can be used again. Once they're inactive, they can't be used. But now crossing that threshold, they can be used again. Our voltage-gated sodium channels close first, but our voltage-gated potassium channels close slower. Because of this slow closing, they do cause the hyperpolarization, what we actually call the overshoot, because it goes past our resting membrane potential. But do you think it reaches its equilibrium potential?
No.
No. So notice neither sodium nor potassium reach their equilibrium potential. Of course, is the cell going to stay hyperpolarized forever?
No.
No. So basically using pumps and leak channels, the cell returns to the resting membrane potential. And now the cell is at rest again. And that is an action potential. Questions on this? All right, we've done this on the whiteboard. Let's look at this with the pretty pictures and the pretty words from your textbook. Remember, we start with the cell at rest, -70 millivolts. Both our voltage-gated sodium and voltage-gated potassium channels are closed. And the cell will happily stay at -70 millivolts forever. However, we then need to cause a disturbance, right? We want to cause a small depolarization to reach that magical point that's not on this simple illustration that reaches threshold. And the way we disturb this is by producing a graded potential on the axon, pardon me, on the dendrites or the cell body. We produce that graded potential. And if that graded potential is big enough to reach -60 millivolts, that's when the magic happens. Again, threshold is the point where all of our voltage gated channels are going to open. But they both open at different rates, right? If grandpa and your teenage daughter, well, now your teenage daughter and your infant, not infant either, adolescent, your 10-year-old are sitting on the couch. Grandpa and 10-year-old are sitting on the couch and you say, go. They're both going to start to get up at the same time, but are they both going to reach their feet at the same time? No, that 10-year-old bounces up, whereas grandpa needs a little bit of time. And that's exactly here. They all hit go at the same time, but they don't all open at the same rates. Actually, I liked that one. I want to leave that there. So let's just erase that. So which one opens first?
The sodium channel.
Exactly. The first one to open are those fast voltage gated sodium channels, which there are a ton of. And which way does sodium move?
Into the cell.
Inside.
Into the cell. So a positive ion entering the cell does what to the cell?
Makes it more positive.
Yeah, it makes it more more positive, which, as Audrey pointed out, was a depolarization. Absolutely. So we get this big, massive, rapid depolarization. And again, it's a big positive signal. That right there is our action potential. Boom. Big rapid depolarization because sodium is trying to get to its equilibrium potential. and we get our big positive signal, our big, huge wave. Now, it's trying to reach its equilibrium potential, but is it going to get there?
No.
No, because two things happen, right? One of the things that happens, as we see here, is that ball of the ball and chain wedges itself into the sodium channel. And what do we call the sodium channel when that ball is wedged in there? Inactive, absolutely. Our voltage gated sodium channel is now inactive. No new sodium can enter. And this is the point when we reach our peak. And what did we say our peak was going to be?
Positive 30 millivolts.
Yeah, positive 30 millivolts. Excellent. Of course, it doesn't stay at the peak because a second thing happens. Our voltage gated potassium channels, Grandpa has finally stood up. And now that those voltage gated potassium channels have opened, which way is potassium going to move? Out of the cell. And a positive ion leaving the cell makes the cell more negative. And so now we are getting that repolarization. However, is potassium going to be happy just reaching the resting membrane potential? No. So it's going to reach the resting membrane potential and keep on going, causing that hyperpolarization, what we call the overshoot. So we've got that. Oops, yeah, get rid of this stuff up here. That hyperpolarization, that overshoot. Now, as we also saw, something important happens on the way down. On the way down, we cross threshold. Notice when we cross threshold, our activation gates close. Our channel, voltage gated sodium channel, is now closed, and that pops the ball of the ball and chain out. It removes the inactivation, which is important because now the channel can be used again. When the channel's inactive, there's nothing you can do with it. It's useless. But now it can be used again. Of course, notice our potassium channel takes longer to close. That's why it hyperpolarizes. But eventually, with a delay, it closes. And thanks to pumps and leak channels, this cell, once this closes, will then go back up to its resting membrane potential. Yes, every time a voltage gated sodium channel opens, that inactivation gate, that ball and chain, wedges itself in there every single time.
Can I get stuck?
No.
No.
It's wedged in there until the gate opens it. The gate pushes it out. So it always inactivates, but when the channel closes, it always pops that inactivation gate out. So yeah. And there you go. Back at rest. So again, your book has these 4 stages. Again, I don't care about the number of steps you use. Just make sure you have all of the events. Event 1 is that graded potential to threshold. Event 2 is the rapid depolarization. Event 3 is the inactivation of the sodium channels and the opening of the potassium channels. That leads to step 4, the repolarization and hyperpolarization. Along that route, we pass threshold, which closes the voltage gated channels, sodium first, then potassium, and then finally the repolarization from this. So again, I don't care about the number of steps as long as you have all of the components. And like we talked about, just like with the muscle tissue, Give me every immediate step. Don't just say it reaches threshold and then you get a rapid depolarization. You have to tell me why. We reach threshold and the fast voltage gated sodium channels open and sodium enters the cell, and there's a lot of it, so we get a big depolarization. Make sure you give me the details. There is one more picture from your textbook that I really, really like. It can seem a little bit confusing at first, but if you look closely at it, I think it's really awesome. Notice here in purple, it gives us and shows us the action potential. But what it shows here in orange and in green are the permeability of the channels. Basically, how much sodium can move or how much it cannot move, right? When you open a channel, permeability increases. So notice when we reach threshold, both channels start to open, their permeabilities go up. But do both their permeabilities go up at the same rate? No. Notice our sodium permeability increases right away. Huge, massive number of sodium channels open. So we get a fast movement of sodium from that fast increase in permeability. But then all the sodium channels inactivate. And when they inactivate, can sodium come inside anymore? Can sodium move?
No.
No. And so notice just as quickly, no sodium can come inside anymore either. So just as fast as sodium's able to come in, just as quickly it's not able to be coming. Whereas with potassium, potassium is much, much slower to open. In fact, if you look at it, when it first opens, it's already on its way back down practically to the point where we reach threshold, and they're about ready to close right again. By the time they open, they're practically reaching threshold on the way down, and now they have to close again. But again, they're slow, much slower to open, much slower to close. So their permeability changes much more gradually. So this permeability kind of shows you the actions of those channels. All right, questions on this? This doesn't make sense. You need to say something now because we're about to add another layer to this. All right. I'm just going to put two numbers on here. So the cell is happily sitting at its resting membrane potential. Then we produce a graded potential, and that graded potential gets it to reach threshold. When it reaches threshold, we get this rapid depolarization, followed by a rapid repolarization, an undershoot, and then back to rest again. That makes sense? Again, I did it super quick, but no new information there. I was just re-emphasizing what we've talked about so far. All right. So remember, as we've talked about, for most work, like even something as simple as lifting my coffee mug to my face, as we talked about, that requires more than one action potential. And let's think about this. What actually produces the action potential? What is the actual event that causes the action potential? An action potential is a big positive signal. So what actually causes that big positive signal?
The sodium channels voltage. Sodium channels opening.
There you go. Bingo. Our voltage gated sodium channels opening. It's like a door to the classroom or the door to my bedroom. And that opening of the channel is what causes the action potential. So where does that opening begin? Where does the voltage gated sodium channels opening start? What causes the voltage gated sodium channels to open?
Reaching threshold.
Yeah. So our voltage gated channels, voltage gated sodium channels open at threshold. If I want to go downstairs during the break and get a cup of coffee, and I open my office door, once that office door is open, can I open it a second time?
No.
No, it sounds
No.
kind of silly, right? So notice once I open these voltage gated sodium channels, because that's what I need to do to get an action potential. Once I open these voltage gated sodium channels, can I open them a second time and get a second action potential?
No.
No. So notice once these channels open, and if I can't open them a second time, I can't get a second action potential. And then something even worse happens up here at the top. What happens here at the top?
They become inactive.
If my voltage gated sodium channels inactivate, can I open them again?
No, not yet.
And if I can't open them again, can I get another action potential?
No.
Exactly. So notice basically what's happening here is I stimulate my neuron. I produce an action potential. And then during this curve, this period of time that I'm looking at right now, we reach a point where it cannot produce another action potential. When we talked about muscle cells, didn't we have a fancy term for a period of time where no matter what you did to the cell, you couldn't get it to fire another action potential?
Refractory.
Exactly. Notice what this is the absolute refractory period. During this absolute refractory period, there is absolutely nothing we can do to produce a second action potential. What do we need to do to be able to produce a second action potential?
Close the.
We have to close the channel. And where would that occur?
At repolarization or threshold.
Yeah. Notice here, when we reach this point, we reach threshold on the way down, our voltage gated sodium channels close. And once they close, now they can be opened again. Right? So if you think about it, if we were to draw a shaded area over this, basically from the time we open, really from the time we stimulate it, from the time we stimulate it to the time we reach threshold on the way down, we cannot get a second action potential. Notice, like we talked about before, this has nothing to do with the first action potential. This is all about when we can produce a second one. And in this red period of time, is there any way for us to produce a second action potential?
No.
No. So this is an absolute refractory period. There is absolutely nothing we can do to get it to fire an action potential again. All right, with me so far? All right, let's look at a different point. Let's say, for instance, this point right here. At this point right here, can I open my voltage gated sodium channels again?
It's technically possible.
Yes. What did it take to open my voltage gated channels over here? When I was over here at this point, what did it take to open my voltage gated channels?
A big positive signal.
Did it take a big positive signal to open those channels?
No.
No, it took a graded potential. And how big did that graded potential need to be?
10 millivolts.
It only took a 10 millivolt change to reach threshold when we were at rest. But what about here at this new point? At this new point, is a 10 millivolt change going to be enough to get us to reach threshold?
No.
No. So notice, technically, it is possible to open those voltage gated channels again, but the same signal we used the first time won't be enough. We can open this channel, but it is going to take a bigger stimulus. So basically, we can produce a second action potential, but it's harder to do. It's not impossible, but it is harder to do. This is what we call the relative refractory period. And the relative refractory period obviously starts after the absolute refractory period, but when would it end? When would our relative refractory period end?
When it returns to rest.
Exactly. Oops, this doesn't need to be in capital. There you go. So again, this has nothing to do with making an action potential, but everything to do with producing a second action potential. We've used this idea of a refractory period. We mentioned it when we talked about muscles, but now because we understand what channels are involved and what ions are flowing, we can actually see those refractory periods and understand why they work. So these are our two refractory periods. Notice I purposely did this on a different page. If I ask you to describe the events of an action potential, do you need to talk about the refractory periods?
No.
No, right? But if I ask you to describe and explain the two types of refractory, identify and explain, or identify and describe the two types of refractory periods or something like that, could you do that as well as a separate essay question?
Yeah.
Yeah, absolutely. Because refractory periods are about producing a second action potential, whereas obviously an action potential is about producing an action potential. And so I've done this here with my pretty pictures. And of course, not surprisingly, your book's got the pretty picture showing those refractory periods as well. The absolute refractory period is the period of time where no matter what you do, you cannot produce a second action potential. And it starts from when it's first stimulated to basically when it reaches the threshold on the way down. And then the relative refractory period is the amount of time where you can produce another action potential. It just takes a bigger stimulus. And again, it's from threshold when our voltage gated sodium channels close till the point where the cell gets back to its resting membrane potential. And your book's got a nice table that talks about graded and action potentials and everything that goes along with that. All right, questions on that? Where are we time-wise? We're doing good. Okay, we can do that. All right, let's go back to this picture. So I'll ask one last time. Any questions on the action potential or the refractory periods? All right, excellent. Now, notice one last thing. As I mentioned, everything we've talked about so far is just happening right here at the axon hillock. Remember, we need to produce an action potential, but after we produce that action potential, we then need to spread that action potential down the axon. And so that's what we're going to do next. Next, we need to spread or propagate the action potential. But we'll do this after the break. We'll give a chance for this information to sink in. Once this information sinks in, then hopefully it'll be easier when we propagate that action potential. So let's take our second break, take another 15-minute break, come back at 10:20, and at 20:20 we will restart, and I will start the recording. All right, any questions? All right, see you in 15 minutes. All righty. We have now made an action potential, and let us now send it on its merry way. We're going to take that picture we were looking at before and change it slightly. Here is the cell body and the axon hillock coming in to the axon. I think we can make this work. And again, the first time we measured it with our voltometer right here. Now, remind me again why it is that our axon is able to produce an action potential, but our cell body and our dendrite is not.
Because they don't have the voltage gated or gates.
Yeah.
I have a question regarding this really quickly.
Yep.
So is this with a myelinated axon or a non?
Ah, excellent question. We are going to start first with an unmyelinated.
Okay, thank you.
Excellent. So our axon has a ton of voltage gated sodium and voltage gated potassium channels. Which ones are the ones that actually produce the action potential?
Sodium.
Sodium.
Sodium. So we're going to put and potassium channels in parentheses here. And just for right now, we're going to focus on the voltage gated sodium channels. And I'm just going to put a few jillion of them here along the length of my axon. And if there was an easier way to do this, I would. And what if I do lines? It's a little faster. Not much. There we go. Excellent. So, as we know, our starting point, how do we produce our very first action potential?
Through the small graded potentials.
Yes. So graded potentials from the cell body and the dendrites reach the axon hillock, and specifically the trigger zone. We don't see the trigger zone, but we know it's in there.
Is this like the first step? Or?
Well, this is how we produce the action potential, right?
Okay, yeah.
So we graded potential from the cell body, came down, wave, traveled down, and reached threshold. So right here at the axon hillock, I have to make this smaller. And when our axon hillock reaches threshold, what happens?
Sodium enters the cell.
Okay, that's the second thing that happens. What's the
always
first
do
thing that happens?
Sorry.
There you
Voltage
go.
gated.
Excellent. Our voltage gated sodium channels open and sodium enters the cell, and we get that big 100 millivolt change. Right, we get our action potential. So our voltage gated sodium channels right here at the axon hillock open. We get a huge, massive influx of sodium. And that produces a big 100 millivolt action potential. Oops. Because all these, actually, do I have a stamp for that? I don't have a stamp for that, shucks. So again, all of this sodium rushes into the cell right at this location, and we have a big action potential. Again, no new information. This is exactly what we've talked about before. However, when all of this sodium rushes into the axon at just this location, is it going to stay just at this location?
No, it's going to spread.
Exactly. So when the sodium enters, our sodium spreads passively. Which way?
Both.
Exactly. In both directions. So when our sodium enters, it is going to spread in both directions. Some sodium is going to go back this way towards the cell body and dendrite. And do we care if sodium spreads back to the cell body and the dendrite? Do we care if the dendrite and the cell body depolarize?
No.
No, they don't have voltage gated channels. They don't have a threshold. So we don't care if it spreads there. But some of the sodium spreads to the next segment. And when sodium spreads to this next segment, it makes the next segment more positive. Probably not a lot more positive, but does it need to make the cell a lot more positive at this location?
No.
No. In fact, how much more positive does it need to make it?
Plus 10.
Yeah. It reaches negative 60 millivolts. And why is that number significant?
It's threshold.
That's threshold. And what happens at threshold?
More gates are open or channels.
Exactly. So in this new segment, new voltage gated sodium channels open. And when these voltage gated sodium channels open, what happens?
Same thing.
Exactly. New sodium rushes in to the cell. We have a massive influx of sodium. That rushes into this area. And as a result of that, we produce another big 100 millivolt change. Now, notice, if we were watching this from the outside world, it would look like our action potential shifted one space to the right. Technically, is that what happened?
No.
No. So it looks like our one action potential has spread, but really what we've done is open new channels, moved new sodium, and made another 100 millivolt change. Unlike that wire in the wall where a single electron moves along the entire length, is it the same sodium ion that moves down the entire axon that makes this action potential spread down the axon?
No.
No, each time it's going to be new. But the advantage of each time it being new is because we're using new sodium and new channels, we're always going to get that 100 millivolt change. So this was 100 millivolts. And this is 100 millivolts. And guess what happens next?
100 millivolt change again.
Well, true, but again, let's walk through the steps. What happens to the sodium that rushed in here?
It's going to spread left and right.
Exactly, our sodium spreads. And how does it spread?
Both directions passively.
Now, this sodium spreading in both directions could cause a problem. This sodium spreading back this way, isn't that going to open these channels and cause another action potential? And suddenly we're having an action potential bouncing back and forth and back and forth?
No.
Why not? Ah, excellent. Mitch has got it. Absolutely. Notice this sodium that comes back. Exactly. The sodium that travels backwards does not trigger an action potential because of the refractory period. So essentially, again, the sodium going backwards has no effect, but it spreads in both directions. And when it spreads into the next direction, then it is going to reach threshold. And when we reach threshold, new sodium channels open. And when those new sodium channels open, new sodium rushes in. And we get another big action potential. Oops, forgot all the other stuff. Open, 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 open. In, and another. Oh. There. 100 millivolt change. Basically, what we are doing here we are producing small local currents. Oops, that's too big. That travel down the entire axon. So again, this sodium comes in and spreads, gets it to reach threshold, opening new channels, sodium rushes in, and we get another action potential, which spreads. And when it spreads, it opens more channels and sodium comes in, and we get another big action potential, which spreads. And as it spreads, it opens the next one, and then it spreads, and that opens the next one. And notice around and around and around she goes. All these local currents causing local depolarizations, causing local voltage gated sodium channels to open. And in this fashion, I know my drawings have gotten smaller, but the key to this is at the end of this whole process, we started with a 100 millivolt action potential, and we reached the end of the axon with a 100 millivolt action potential. The signal never gets smaller. So like I said, it's like producing a wave in the pool that travels the whole length of the pool and never gets smaller. And unlike the wave in the pool, the reason is because it is always, and here's the fancy term we use for it, it is always self-strengthening. We are always adding new sodium to opening new channels and adding new sodium. So that the signal never gets smaller. And so you can see, as I mentioned, I could have a neuron that went from here all the way to Seattle, Washington. And I could produce an action potential here, and it would get all the way to Seattle, Washington. Now, would the same sodium that enters here at the axon hillock be the same sodium that reaches the end in Seattle, Washington? No, but we would be able to get a 100 millivolt signal all the way from here to Seattle, Washington. Again, if you don't like my pretty pictures, let's look at the pretty pictures from your textbook. Again, here we have the trigger zone. It reaches threshold, sodium rushes in. That sodium rushes to the next area, producing that local current, which depolarizes the next segment to reach threshold. And when it reaches threshold, it produces an action potential. Massive amount of sodium rushes in, and then that process continues. This is a beautiful, elegant system that, like I said, could carry an action potential, could carry a signal all the way from here to Seattle, Washington. It's an awesome, awesome process, but there's one minor problem with it. Back in ancient times, there were things called telephones, right? And by telephones, I don't mean the thing that's in your pocket that takes videos and takes pictures and allows you to watch the internet and all the kind of fun things that goes along with that. I'm in these big boxes that were connected to the wall with big, huge rotary dials on them. And if your grandma happens to live in Seattle, Washington, right? Well, first of all, hopefully she's not drowning right now. But if she lives in Seattle, Washington, in ancient times, you'd be able to pick that phone up off the wall, dial those 10 numbers into that rotary dial, and grandma would pick up the phone on the other end, and an electron, excuse me, give me a second so I can finish my rant. An electron would carry my signal. I would say hello, and an electron would carry that signal all the way to grandma's phone in Seattle, Washington. And how quickly did grandma get that signal? From here to Seattle, Washington.
Quickly.
Yeah, pretty much instantaneously. I would say hello, she would say hello back. I would say how you were doing, she would say I'm fine. And we'd pretty much have an instant conversation. Now, again, in ancient times, if grandma instead was in England, was it possible to call grandma on the phone when she was in England?
No.
Well, okay, ancient times, no. But then governments got together and put these massive cable lines that run across the entire length of the ocean. We have these big, huge cables that run across the entire length of the ocean so that that electron could actually get from here to England. Now, that electron basically moves at the speed of light practically. But when I'm here in California and grandma's in England and I say hello, does she get the signal instantaneously then?
Yes.
Well, it was a couple seconds. So, I mean, it was pretty instantaneous, but it was a couple seconds. So I'd say hello, and 5 seconds later, I would hear back hello from her, right? There was a little bit of a delay, but we could still hold a conversation. If instead, my phone used an axon to carry that information from here to Seattle, Washington, I would say hello, and my axon can carry that signal from here to Seattle, Washington. But instead of being instantaneous, it would take 2 days for that signal to get to Seattle, Washington. So I would say hello, and 2 days later, grandma would hear it. She would say hello, and 2 days later, I would hear that. I would say, how are you? 2 days. I'm fine, 2 days, right? Grandma's going to be dead before we finish this conversation. So while this is an incredibly important and useful process, as we talked about, this is an unmyelinated axon. So we want to be able to speed things up. So what can we do to speed this up?
Some myelin sheath on it.
Exactly. We can myelinate these axons. And remember, myelinating them can make them 5 to 6 times faster. Now, remember also, we said most axons are myelinated, but not all. How can we use an axon that would take 2 days to send a signal to grandma in our body if it's so slow?
Because we're not going to Seattle or wherever grandma is. It's in our
Right,
body.
absolutely. You don't have axons in your body that are long enough to reach Seattle, Washington. You have axons, the longest axons in your body are 3, 4 feet long at most. So is it instantaneous? No, but we're still talking about fractions of a second, right? In fact, you can measure it. In the classroom, we actually have some measuring sticks you can use. And now that you're on the interwebs, there's all sorts of reaction time quizzes you can take, websites where you stare at a red light, and as soon as it turns green, you push the space bar, and it tells you how quickly you reacted. A lot of that delay in reaction is the travel of that action potential. The action potential from seeing the light change has to go to the brain. You then have to process that information and send the signal out to your hand, telling it to click. So some of that delay is from that axon. But again, we're talking, a good reaction time is 150, 200 milliseconds. A bad reaction time is 500 milliseconds. I mean, we're still talking fractions of a second here. So yeah, the real key is that the axons we're using are much, much shorter, but we can still make them better. So let's make them better. Here's my axon again with my voltage gated sodium and potassium channels. But again, remember, we're primarily just focusing for propagation. So this is propagation of an action potential along a myelinated axon. And remember that myelin is basically a big, huge chunk of fat. Hundreds of layers of phospholipid bilayer wrapped around the axon. And someone remind me again, what cells are responsible for producing the myelination?
Schwann cells and like dendrites or dendrocytes.
Dendrocytes. There you go. Excellent. Where do the Schwann cells myelinate axons?
The peripheral nervous system.
Excellent. And the oligodendrocytes do it in the central nervous system. Excellent. They take their plasma membrane and they wrap it hundreds of times around the axon, forming that big, thick layer of fat. Now, think about it. If I have a big, huge layer of fat, would there be much of a point of putting an ion channel on the axon in that location?
No.
No, because there'd be nowhere for the potassium to go, and there'd be no way to get sodium in either. So we're not going to put them there. However, does the myelin cover the entire axon?
No, there's nodes.
Right, exactly. There are gaps in between those nodes of Ranvier. And it is in those nodes that is where we find our voltage-gated sodium and our voltage-gated potassium channels. And I need to cheat and move this. Move it over here. All right. We comfortable with the anatomy? Excellent.
Yes.
So again, starts the exact same way. We produce an action potential and it spreads to the axon hillock. When it gets to the axon hillock, our axon hillock reaches threshold. Our voltage gated sodium channels open. And sodium rushes in. we get our big positive 100 millivolt signal that we call our action potential. Exact same thing that happened last time. Oops. All right. And again, all this sodium rushes in. And just like before, our sodium spreads passively. Some of it is going to spread back to the cell body and dendrite that we don't care about. And some of it is going to spread down the axon. But this is a passive spread, like the wave in our pool. And as this spreads passively, what happens to a wave as it spreads passively? Well, okay, let's think of it this way. Let's say 10 sodium start traveling down the axon. Are all 10 going to reach the far end?
No, it's going to decrease.
Yeah, we're going to decrease. So then maybe at the next place, there only 8 make it here. And then in the next spot, maybe only 6 make it here. And maybe only 3 make it here. As it spreads passively, the signal gets smaller. We call this attenuation. Now, there is an important reason for this. When a charged particle, like a sodium, I'll cheat and do this here, travels, when it's in the axon, there's basically 2 things it can do. It wants to either move across the membrane, and clearly it can't do that when it's here inside of the, underneath the myelin. But remember, sodium is a charged particle. So if there is a negatively charged ion on the outside, that positively charged ion can actually be attracted to it, where it'll basically, right, lean against the wall, trying to speak with its loved one on the other side, trying to get together with its negative, with its opposite on the other side. Now, with hundreds of layers of myelin, is it likely to be able to attract a negative ion on the outside? Or do you think it'd be more likely to be able to attract it in a place like this without myelin?
Without myelin?
Yeah, without myelin. So luckily for us, that myelin reduces the attraction. That attraction, again, from a physics standpoint, that ability to basically store a charge on the end, which is what we're doing, what we do is actually it's capacitance. So basically, if you want to get fancy with physics, the myelin produces a reduced capacitance. But it's just a fancy way of saying it discourages the sodium from stopping, right? Basically being attracted to another ion. So more of it spreads, but even though more spreads, it still attenuates. So as it spreads, our signal, like the wave of the pool, gets smaller. It started as a 100 millivolt change. Is it going to be a 100 millivolt change when it reaches the first node?
No.
No. But does it need to be a 100
No.
millivolt change? No. What does it need to be?
10.
Notice it can attenuate 90%. It can go from a 100 millivolt change to a 10 millivolt change. But as long as it's still at least a 10 millivolt change, then what's going to happen?
Reach for threshold.
Yeah. We reach threshold. New sodium channels open. New sodium enters the cell. And we get a new action potential. A big 100 millivolt change. And guess what happens to this 100 millivolt change?
Decreases as
Yeah.
it spreads.
Again, it passively spreads and attenuates, getting smaller as it goes. But as long as it is at least a positive 10 millivolt change, then again, the process continues. Or again, it'll reach threshold, voltage gated sodium channels open, sodium rushes in, and we get another big 100 millivolt change. This type of propagation is actually what we call saltatory conduction. Saltatory is just a fancy way of saying, or what it means is jumping. Because if you think about it, it looks like the action potential is jumping from node to node to node along the way. And again, if you don't like my picture, we got the pretty picture from your textbook. Again, showing that saltatory conduction. Again, that's just a fancy way of saying propagation with myelin. At the trigger zone, we get our action potential. It spreads passively under the myelin, attenuating as it goes. And as long as it reaches threshold at the next segment, it triggers the opening of new voltage gated sodium channels, new sodium rushes in, and that process continues. As I mentioned, the advantage of this is that it is much, much faster, 5 to 6 times faster than just a simple unmyelinated axon. And as I mentioned, not surprisingly then, most of our axons are myelinated. But what's the key word in that statement?
Most.
Because most means not all. In fact, some of the nerves that aren't myelinated are your pain nerves, the nerves that carry a signal about pain to your brain to tell you you're in pain. Don't you want to know you're in pain as fast as possible? Wouldn't it make sense to myelinate those?
No.
So why aren't they myelinated? Well, you say no, why? Why wouldn't you want them to be, why wouldn't you want to know that you were in pain as fast as possible?
Well, you would, but this process isn't the best way to do it because you're banking on the fact that it can jump and reach that 10 millivolt threshold. Therefore, it's not consistent and not really reliable.
Exactly. This method does have a risk. That signal has to be strong enough when it reaches the node to reach threshold. And if it doesn't, then that signal fails. So yes, the pain pathway is a little shorter, but it is 100% reliable because the signal doesn't change size. Here, the signal changes size. And because it changes size, there is a risk to that. We actually talked about it in the last class. If you remember, one of the things we talked about is there is an autoimmune disorder where your body's own defenses, your own defense cells start breaking down the myelin. When they break down the myelin, that lessens the size of the myelin. And notice now more sodium can be attracted to ions outside. So our signal attenuates faster. And when it reaches the next node, it may not be big enough to reach threshold. What did we call that condition?
Multiple sclerosis?
Exactly. We called it multiple sclerosis. Multiple sclerosis is a central nervous system autoimmune disorder where our own immune cells, our own body defenses, attack and destroy oligodendrocytes.
So is it an autoimmune disorder?
Yeah, autoimmune. Yeah, our own body's immune system attacks us. So this is an autoimmune disorder. Absolutely. All right. However, diseases are not the only thing that can stop this signal. What happens? All right, you're playing baseball, you're up at bat, bases are loaded, right? And it's the bottom of the ninth, score is tied, and the pitcher throws that 105-mile-an-hour fastball, and it hits you in the arm. And you walk to first base. What do you do when someone hits you in the arm?
You rub your arm?
Yeah, you rub it. Why do you rub your arm when someone hits you?
Kind of takes the pain away.
Yeah, it feels better. Doesn't it feel better when that happens? Do you know why? I don't either. No, I'm just kidding. The reason why is that your tactile receptors of the skin, when you rub the skin, that tactile sensation from the skin sends a signal to your brain that you're being touched. And that actually confuses the pain signal. And so as long as you're rubbing it, you're confusing the pain signal and you don't recognize the pain signal. Of course, as soon as you stop rubbing it, the tactile sensation's away and then the pain sensation comes back. So notice there are other ways we can disrupt that signal from getting to the brain. Rubbing the arm is one way. Toxins are another way that we can do this as well. If we can block the voltage-gated sodium channels, we can stop that signal from getting through. When you go to get that root canal, what does the doctor inject into the nerves to block that pain signal from going to your brain?
Novocaine?
Novocaine, absolutely. Lidocaine is in that ointment you put on your arm when you get home because it's sore, or on your back after a hard day of work. So things like Novocaine and lidocaine, those are local anesthetics that can block voltage-gated sodium channels. You probably don't have Novocaine in your medicine cabinet, but you probably have some form of lidocaine in there. Absolutely. Let's talk about another really interesting toxin, tetrodotoxin. Tetrodotoxin, which is called TTX, is that toxin that is found in pufferfish. Now, like I said, you're not going to find that in your medicine cabinet here in your house, but where is one location where you might find pufferfish toxin?
Pufferfish.
True. And where might you find pufferfish?
Like, right, if it's not in the United States.
True, you're correct. Not in the United States. Used to be in Canada. You used to be able to get it in sushi bars in Canada, but now I think it's illegal in Canada as well. However, in some countries, in some sushi bars, are you able to get a specialty called fugo?
Yes.
Yeah, absolutely. Like I said, it used to be legal in Canada. However, in Canada, the chefs that prepared it, they had to be certified every 3 months. So every 3 months, they had to take another test for their certification. When you went to the restaurant, you had to sign a 5-page waiver before they would serve you the dish. But the goal is to have the chef talented enough where he was able, because basically they have a sack that contains the tetrodotoxin in them. And the goal is to cut the meat in such a way that it has the tiniest bit of tetrodotoxin on it. It's a long-lasting voltage-gated sodium channel blocker. so that when you pick it up with your hands, you get a little tingling sensation in your fingers. When you put it in your mouth, you get a tingling sensation on your tongue and on your lips. However, not so much tetrodotoxin that it gets into your blood and goes to places like your diaphragm and stops you from breathing. You know, I think something like 8 people a year die from improperly prepared fugo. Like I said, it is completely illegal in the United States. It is now illegal in Canada, but there are other places in the country where you still can get it. It's used for research a lot. My research on the visual system, we use tetrodotoxin, and it is a very dangerous toxin that there are a lot of regulations on its use and all these kind of things like that. But again, it is another example of these voltage gated sodium channel blockers that we use to stop action potentials.
I have a question.
Yes.
So
It tastes like fish.
yeah, I know. It's okay. I was wondering, when your body goes into shock, does it stop? Like, let's say you, I don't know, you have a compound like fracture and it goes through your skin. Like, and I've had a friend who did that, and then she said she didn't really feel it after a while. But does that have anything to do with these voltage gated channels?
No, that type of shock is more of a higher order cognitive function, where basically the pain signals become so overloading to the brain that the brain basically habituates to it and disassociates from it. So it's more, so shock, that kind of shock is more of that type of a thing.
Oh, okay.
So it's more of a higher order. Again, what you have to remember is you are being constantly inundated with information from your skin, right? Pain, temperature, tickle, itch, pressure. Like I said, if you really stop for a second and think about it, you can feel the socks on your feet. You can feel your chair on your back and on your butt. You can feel the weight of your shirt on your shoulders. All that information is constantly coming into you. So those signals are going into your brain constantly. However, you're not always thinking about the weight of your sleeves on your arm until someone like me points it out to you. But before I said anything, it was there. You weren't paying any attention to it. So one thing is how the signals get in. Voltage gated sodium channel blockers stop the signal from getting to your brain. However, how we process that information once it gets to the brain is a completely different type of process. And so that type of shock has nothing to do with getting the signals to the brain and everything to do with how our brain processes it. Now there is, I can't think of the name of it, there is a medical condition where your pain neurons basically don't send signals to your brain, or your brain doesn't process those signals. And those are the people who, you know, can accidentally cut themselves or burn their hand and they're not completely aware of it. There's a name for that condition. I can't think of what it is, but yeah, that would be have more to do with the signals going in. But it's more than just that. All right. So there you go. We've made an action potential, we've propagated an action potential, and now we've even modified that action potential. Excellent. And propagated it both with and without myelin. Those all sound like good potential essay questions to me. All right. Questions on that? Yikes. Promised, I told you we'd be getting behind. All right, excellent. Not done. We're done with lecture, but what we need to do now is we need to shift gears into lab mode. So let's bring this back up. So as I mentioned, we are going to be going, oh, that's not what I wanted to share. That's what I want to share. We are going to be going through our nervous system, central anatomy list. So it's the gross anatomy of the nervous system list. We've already kind of started talking about those things. So we'll go over the structures again briefly, and then from there we'll get into the lobes and all of the information about that. So we will do that, but we'll do that after the break. We'll take one last break. We'll take a 15-minute break. So that looks like we will restart at 11:20. All right, and I'll start the recording at that time. All right, any questions on that before we switch gears? All right, one last chance to stretch, go to the bathroom, get something to eat, get something to drink, cry, whatever you need to do to make it through the rest of the class. Get that done, and then we'll meet back here at 11:20 and start talking about some of the fun stuff. See you then. All righty. Let's go ahead and get started. Again, we are working our way through our nervous system gross anatomy list. We are starting with the telencephalon. And again, remember the telencephalon is that first cephalon, and it's basically this entire mitten-like structure that is the largest component of our brain. As we talked about in the last class, the tissue is folded in on itself. And what is the name we give the raised ridges?
The folium.
Remember the folium down here in the cerebellum,
Oh,
but what do we call
okay.
the raised ridges on the cerebrum? What do we call the raised ridges on the
Oh, maturity
cerebrum?
answer. That's why I was confused.
Oh, I'm sorry. No, I didn't see that. Excellent. Gyrus, excellent. And what do we call the grooves?
Fissures.
Well, the typical groove is a sulcus, but you are right. A very deep groove is known as a fissure. So again, as we were talking about, if we were to cut this and look at the side, right, a small indentation is typically a sulcus, whereas a very deep indentation is a fissure. Those are generic terms, but remember we also had some specific names for some of these. This specific sulcus that starts at the front of the head and comes down like this, what did we call that one?
Lateral. Lateral.
No, what was the one that basically starts at the top near the center of the cerebrum and comes down?
Oh, is it the longitudinal?
No, not a bad
Okay,
guess.
then I don't know.
Well, there you go, central sulcus. Absolutely. You should have the list in front of you, so it should help with that. Excellent. The raised ridge in front of the central sulcus, what did we call that?
Precentral gyrus.
Gyrus, absolutely, gyrus. And again, as long as you spell it correctly, I don't care how you say it. And the raised ridge behind the central sulcus, what was that?
Postcentral
Postcentral gyrus.
gyrus.
Yeah, postcentral gyrus. Excellent. Perfect. Excellent. Here we see that in that sagittal section of the brain. We can see those areas that way. Notice also that like the cerebellum and unlike the spinal cord, the cell bodies and dendrites are located at the superficial part of the cerebrum, what we call the gray matter. Whereas, and in this case, that gray matter collectively is known as the cerebral cortex. Whereas the portion that is comprised of the axons is our white matter. And again, remember in our brain and spinal cord, that white matter we call tracts. However, I do want to show you something. Notice they've made these very colorful looking components. The pink and the light pink and the dark pink and the purple. These structures down here are also gray matter. They are also clusters of cell bodies. And these clusters of cell bodies in the central nervous system that are not part of the cortex, we call a nucleus, singular. So I guess I should make that singular as well. A cluster of cell bodies in the central nervous system, we call a nucleus. So these are all examples of nuclei, which would be the plural. So notice for our gray matter, we have cerebral cortex and nuclei, and our white matter forms tracks. And most of our gray matter, especially the cerebral cortex, is superficial. Oops, didn't I spell superficial correctly? Our white matter tracks are deep. However, notice our nuclei are going to be deep as well. All right. Now, go ahead.
So I had a quick question.
Yeah.
So on our nervous system histology, I had a few questions and I was trying to draw them yesterday. So if you point, for example, to the gray matter, it's also called cerebellar cortex. Will you use specific language when you want us to answer it either or? Like I'm guessing if you point to the matter and ask what matter this is, we'd say gray.
Yeah.
But
so if I were to point with this arrow, I could ask you to identify the type of tissue or something like that, and you would say gray matter. However, if I were to ask you to identify this entire gray matter structure, what would this entire gray matter structure be?
the cerebellar cortex?
Yeah, exactly.
Okay. So it's just the entire
So again,
structure.
careful. Cerebral cortex.
Oh,
Cerebellar
cerebral. Sorry.
cortex is the gray matter of the cerebellum.
Okay.
So this is the cerebral cortex.
And then I had one more question about the histology.
Yep.
You didn't really go over the cell body. So what would you ask? I guess, would that be considered like a structure or because that's on our histology for a lot of them, but I'm not exactly sure.
Region of the neuron, region of the cell,
Okay.
cell body. Yeah. Basically, it's just to delineate it from the processes that stick off of it.
Right.
Right. All right. Notice again, when you look at the white matter tracks, remember these are axons carrying information from one location in the brain to another. Which again, if you think about it, makes sense. If I am manipulating an object with my hand to try to figure out what it is, does what my index finger and my middle finger are touching, do that information need to be coordinated to figure out what something is? Yeah, so we can have little local connections for stuff like that. However, what I'm feeling and what I'm seeing, do I need to connect that? Absolutely. And so I can have longer tracks for that. And like at the end of class every day, when everybody gets on camera and stands up and applauds after I've had another amazing lecture, do you need to be able to coordinate the activity of the two hands for your left hand and your right hand to be able to do that? Absolutely. And so you need tracks that go from one side to another. And these tracks that go from one hemisphere of the brain to another are what we call commissures, which we will have several of. So I wanted to mention that here. All righty. Lastly, as we mentioned here, we see again a real brain, and there are 4 lobes. Those 4 lobes are delineated by the bones that protect them, but there are also some anatomical features that distinguish them as well. Notice the brain is actually divided into 2 hemispheres by a big, large groove known as the longitudinal fissure that goes right down the midline of the body. Notice if we cheat and go back to that gray matter, white matter picture, notice here we also see that longitudinal fissure. That big, deep invagination that separates the brain into 2 hemispheres. However, notice it doesn't completely. The brain is connected in the middle. So it doesn't completely dissect the brain in half, but we have these 2 big, large lateral hemispheres, the left and the right hemispheres that we see separated by that longitudinal fissure. We also talked about the central sulcus already. Notice the central sulcus separates the frontal lobe from the parietal lobe. We keep talking about the cerebrum looking like a mitten where you have the fingers separate from the thumb. And notice there is a very deep invagination that separates the temporal lobe from the frontal and a bit from the parietal. And of course, because it's a very deep invagination, it's called the lateral fissure, right? No, it should be the lateral fissure, shouldn't it? But what do we know?
Anonymous.
hate us.
Status, absolutely. And this is another example of that. Excellent. So we have our frontal lobe, our parietal lobe, our temporal lobe. And notice on the surface, there is no anatomical features, but there are some deep anatomical features that separate the occipital lobe from the others. They're just not on your list, so you're not responsible for them on the gross anatomy. But there are, trust me that there are. And that separates the brain into the 4.5 lobes that we have: frontal, temporal, parietal, and occipital. All right. Questions on that?
I'm not worried about it being like super shiny fucking, you know, like giant glimmering and shit.
Yep, I agree. So let's go ahead and mute all that. You feel free to talk when you need to. Excellent. And again, here we see the pretty picture showing that. And lastly, here we see that again, we see those nice divisions of the cerebrum. Although, like I said, I think this one's probably the best one. So as we mentioned now, we've identified our lobes, but within these lobes, there are certain regions or areas that have specific functions. And so that is what we need to focus on now. Now, again, this is a really good illustration, but let's kind of simplify this a little bit. There is our, I think I can make this work. Maybe cheat a little bit, make it a little bit bigger. There we go. There is our mitten-shaped brain. And as we know, we have the lateral sulcus, which we've drawn, and we'll go ahead and put the central sulcus in here as well. And then that will allow us to identify, man, I really did a bad job of this. That'll work. Our 4 lobes. And let's talk first about our frontal lobe. In our frontal lobe, how many areas are you responsible for?
3.
3, excellent. And what is the first one?
Prefrontal cortex.
Prefrontal cortex, as the name indicates, is basically the very front part of the brain. So this is the part here. Let me see if I've got a good picture that I want. Ah, I do. Excellent. This is what I want. All right. That one can cheat now and go back to this. So we have our prefrontal cortex. Our prefrontal cortex is basically the part that is association cortex. This is where our intellect, this is where our personality, this is where our reasoning is located. How do we know that?
From injuries.
Yeah, absolutely. One of the greatest things that ever happened to the field of neuroscience was the development of helmets. Because before helmets, young men went away to war and they died. After helmets, young men went away to war and they came back with only parts of their brain blown off. And they survived those injuries. And as a result of that, we saw what part of their brain was missing. We saw what they weren't able to do. And that kind of helped us to figure out what it was that the brain did. And this prefrontal cortex is the best example of, is a great example of this, right? Yes, people got injured in war, but probably one of the most famous cases didn't happen in war, but happened to a young gentleman by the name of Phineas Gage. Anyone here know who Phineas Gage is? Excellent, Mitch. Who's Phineas Gage?
He got like a rod from a railroad driven through his frontal cortex.
Exactly, absolutely. Phineas Gage was a foreman on the railroad. Foremen had to be very emotionally stable, mature individuals because they were responsible for dealing with the dynamite, right? When you're building a railroad and you happen to come across a hill, do you take the time to build your railroad around the hill?
No, you blew it up.
No, you blew it up and then kept the railroad going straight. So the foreman, what they would do is they would drill a hole down to the appropriate depth. And then the foreman would put in the dynamite, and using this 10-foot tamping rod and a little hammer, he would push the dynamite down to the appropriate level in that hole so that when it blew up, it exploded the rock and they could keep going. Well, one day, good old Phineas was doing his job and the dynamite went off. And that tamping rod came out, hit him in the forehead, came out the top of his head, flew 20 feet in the air, knocked him back, and remarkably, he survived. He lived from this, but the front part of his brain was destroyed. And it changed his personality dramatically. As some of the records state, he went from an individual, like I said, who was very stable, very mature, to the type of woman, to the type of man who would drink during the day and curse at women. It completely changed his personality. Right. And then, like I said, others got injured, and then they did some animal research where they'd have these really aggressive monkeys, and then they would destroy the front part of their brain and they would be placid. They wouldn't be aggressive anymore. This was a huge, remarkable finding at the time. We had found the part of the brain that controlled your personality. And if we didn't like your personality, then maybe we could change it. Right? Maybe you were too aggressive. Maybe you were a woman who felt that women should be allowed to vote. Maybe you were so aberrant that you were a male who liked other males. Right? If you had some type of aberrant behavior, we had a way to modify it. Right? Basically, what they would do is they would take a metal rod, and this metal rod they would basically insert into the nasal cavity. And as they would insert into the nasal cavity, they would push it through the cribriform plate, or sometime they'd go through the orbit of the eye. But the goal was to get this hollow tube into the front part of the brain. This hollow tube had a little bit of a slit in it. And out of that slit, they would push a piece of razor wire. And this, oops, hold on. This razor wire would bow out of this slit. And basically that would rotate that around, cutting the connections in the prefrontal cortex until the person's behavior changed to what we wanted it to be. And what did they call this process? Yeah, frontal lobotomy. These frontal lobotomies, oops, did that wrong. These frontal lobotomies were done on a conscious individual. They would bring the person in. Not surprisingly, they would be very nervous. They would insert these cannellini into their brains, and they would continue to rotate it until the person wasn't nervous anymore. There were some, I hesitate to use the word doctors, who would roam the countryside doing these. One of the biggest was an individual by the name of Watson. He did thousands of these. He actually had 2 of these cannuli that he would keep in holsters on his sides, and he would do both nostrils at the same time. He would go to different hospitals around the country and would compete for speed against some of the other local doctors to see who could do them faster. Right? Hopefully, you are appropriately appalled by these behaviors. Right? But you've got to also remember the world that they lived in at that time. If someone had a mental disability, they were put in a sanitarium. And what was the job of that sanitarium? To make them better? No. Their job was basically just to house them until they died. Exactly, isolation. Keep them from hurting themselves and hurting anybody else, and that was it. So the idea that we may be able to make someone better and allow them to leave that sanitarium and go back into society was, you could argue, a noble cause. Now, what's the problem with this process? Aside from the fact that it's brutal and hideous, what was the other problem with this process?
Would you be a vegetable after?
Right. Well, yeah, it didn't work. It didn't always work. Did some people get better from it? Yeah. Did other people not change at all? Did other people get worse? Absolutely. So there was no consistency, right? It didn't affect everybody the same way. And of course, there was always the side effects. You're inserting a tube from the nasal cavity into the brain. Did they have good antibiotics or even understanding of antibiotics at that point in time? So there were tremendous infections, cerebral spinal fluid draining into the nasal cavity, causing problems, all sorts of complications and infections and all sorts of issues that could be caused by this. And as gruesome, as unsuccessful as it is, thankfully, they finally stopped it because they realized what they were doing was wrong and that it wasn't effective, and that's why they stopped it.
Probably not.
No. Why did they stop it? Why don't we have frontal lobotomies today?
Because we figured out.
Well, maybe, no, again, protesting is definitely a very new thing. So, well, not entirely new. I mean, women's suffrage, stuff like that. But again, to the level, they didn't have social media the way they did this way. And again, as we know, people always do what is right all the time, right? So that's why they stopped. They realized that it was wrong. It was bad. And that's why we stopped doing things.
No.
Is it because we got rid of the asylums?
No, I mean, there's still mental hospitals, right? We don't necessarily call them, right? Legislation. You guys are adorable. I appreciate your optimism. You guys are absolutely adorable with all of your optimism. No, they found something cheaper and easier, right? And what's cheaper and easier than a frontal lobotomy? Pills, right? You don't behave the way I want you to behave. What do? I give you a pill. I may not know what that pill does, the mechanism by which it works, but for some of you, it makes you behave in a way that I want you to behave. Of course, it doesn't work the same for everybody. So I give person A one pill, I give person B a different pill. Person C gets both pills. And I just keep randomly giving you different pills until you finally behave the way that I want. I don't know how the full mechanism by which these pills work. I don't know the long-term implications of these pills, but as long as I can get you to cheaply and easily behave the way I want you to behave, then that's all I care about. Plus, it makes my shareholders at the pharmaceutical company happy. I truly hope that 20 years from now, we will be as appalled at the medication of society now the way we are at these frontal lobotomies from 30, 40 years ago as well. Because what we're doing is really just as brutal, just as dangerous. But it's cheap and easy, and it doesn't look gross. So that's why we do it. All right. So yeah, for all your highborn stuff, we're not much better than we used to be. We're still trying to find ways to modify people's behavior. We've just found a cheaper and easier way to do it. Right? When I was growing up, or I guess luckily I missed it, so slightly after, but ADHD, right? Every other kid was on Ritalin for a while, right? Luckily, that's gone down a bit since then. Again, this is not new behavior, and it is still continuing. We're just doing it in a different, less gruesome way. All right, I will get off my soapbox now. Questions on that? All right, so let's go back. Oh, we are here. Never mind. Go back to this picture. Nope, this picture. Excellent. All right, let's talk about the next area that I want to talk about. Notice on the frontal cortex, there are these 3 parallel gyri. These 3 parallel gyri, notice Brodmann called 44 and 45. This is what is known as Broca's area. Of course, why is it called Broca's area?
Broca found it.
Bob Broca found it.
Yeah, good old Bob Broca was the first person who identified them and described it. Let's move it over here a little bit more. Luckily, it is very distinct and very easy to see. So good old Bob Broca was the first person who identified it. However, it has been given a new name as well. Broca's area is also now more appropriately referred to as the speech area. And guess what the speech area does?
Responsible for your speech.
Produces speech, absolutely. Again, just like we talked about with the prefrontal cortex, which we didn't label.
Yeah, I'm confused. So if you ask us, okay, what is what does this area or region do? You would say it's responsible for your intellect, personality, and reasoning.
Yeah, exactly.
Okay.
So on the exam, if I showed you this picture and I had an area basically, oops, where's my arrow? I had an area going here. Identify the region or area, you would say.
Prefrontal cortex.
If I asked you identify the function of this region / area, you would say.
Intellect, personality, and reasoning.
Yeah, there you go. If instead I moved my arrow here, identify this region or area, you would say.
Broca's area.
Yeah, or speech, either be acceptable. And what
Okay.
does it do?
It's responsible for your speech.
Producing speech, absolutely. Exactly. So that makes it easy. The speech area produces speech. There's a couple interesting things about this. For starters, this is one of those examples of the lateralization that we talked about. For most people, over 80% of people, Broca's area is found on the left side only. How we know this is actually kind of cool, right? Again, nowadays we have these functional magnetic resonance imaging machines where they can shove you in a tube and they can look at different blood flow to different parts of your brain. So they have you talk and they see more blood going to this side of your brain and they can tell that that's which side it is on. However, in ancient times, they weren't able to do that. But for the most part, the blood flow to the left side of your brain and the blood flow to the right side of your brain is separate. They intermix a little bit, but for the most part, it's separate. So what they would do is they would inject an anesthetic into your common carotid artery that would anesthetize the left side of your brain, and then they'd ask you to say your name. And if you couldn't say your name, it's because that's the side that your Broca's area was on. So if this is on the left side, any idea what might be on the right? It tends to be more computational skills, more math-related skills. The other interesting thing about the speech area is producing speech is a very, very specific process. And there's a couple ways that we see this, right? Adele has been all over the news recently. I'm not sure exactly why. I haven't been paying attention that closely. I don't know if she's got a new album coming out or she got married. I heard she lost weight. I don't know, whatever it is. She's been on the news a ton recently. And while I haven't bothered, I assume it hasn't gotten any better. Has anybody listened to her ever in an interview before?
Yes.
Can you make out what she's saying without the subtitles when she's talking?
Yeah.
Okay, what, easily?
No.
Okay, she has an incredibly thick accent. But what about when she sings? Does she have that same thick accent when she sings?
No.
No, because speech and singing are two completely different processes. So even though when she talks, at least to me, it sounds like she's got a mouthful of marbles, she sings and it is crystal clear. Right? Someone who has damage to this area can't produce speech. So instead of saying me and Billy are going to the store to get milk, if I had severe damage to my Broca's area, I either couldn't speak at all, or maybe I'd be like, me, Billy, milk, right? None of the cadence, none of the prepositions, none of those kind of things. And even people with minor problems with Broca areas can have some difficulty talking. What do we call those types of people? Or what do we call that condition? Stuttering. One of the ways that stutterers can deal with the issue for certain types of stutters is that they can actually speak in more of a sing-songy kind of voice. Instead of stumbling over the words and stuttering, I can say, me and Billy are going to the store to get some milk. By talking in more of a sing-song fashion, because singing and speech don't occur in the same location, many people with stutters can overcome their stutters by talking in more of a sing-song fashion. So again, it kind of shows you the beauty of how specific and elegant some of these regions are. This is solely, precisely the production of speech. All right. Questions on that?
Clarification's only on the left side.
For 80% of people, yeah, it's going to be on the
Okay.
left side. Absolutely. And then on the right side tends to be more computational information. All right. The last region, and again, this is where things get a teeny bit tricky. So let's do this. There is a structure over here in front of the central sulcus. What is the structure that is in front of the central sulcus?
The precentral gyrus.
Excellent. It turns out on the precentral gyrus is the third area or region that we are responsible for. So on our precentral gyrus is a region or area that is known as the primary motor cortex. And with a name like primary, oh, I'm gonna have to move this now. With a name like primary motor cortex, what do you think it does? Excellent. You get partial credit for motor function, but at this point in the class, we can be more specific. What type of motor function?
Somatic.
Excellent. Okay, so what does that mean? What
Your
is
body.
what is somatic motor control?
Your body.
What parts of your body? There we go. Skeletal muscle. All right. What kind of control is it?
Voluntary.
Voluntary. Excellent. So very voluntarily controls the skeletal muscle. If we're looking at the left side of the brain head, like we are here in the left hemisphere of the brain, what skeletal muscle would the left side of the brain control? Right side. And remember, there's a fancy word for that, contralateral. Isn't it fun when all the information we use gets put together? Exactly. Our primary motor cortex provides the voluntary control of the contralateral skeletal muscle. So over here on the left side of my brain, I can control my right hand to wave to you. And in fact, there's a whole map of the body here on this. Down here is the face, then comes the hand, then comes the arm, and then the trunk, and then the leg, and then right up here at the top, the feet, and then around the corner down the sulcus, we have the genitalia and the internal organs and stuff like that, like the tongue and things along those lines. Handed dominant, so great, that is an excellent question. So here are the things that I will say about that. Your preference in your dominant hand is genetically controlled. However, the other issue with that is there are strong social cues that also can affect and influence that. My mom was actually born in Portugal, lived there for the first 7 years of her life. And in Portugal, if you are left-handed, that is considered a sign of the devil. So when she was at school, which of course was at a church, and anytime she tried to hold the pen with her or pencil with her left hand to write, what would the nuns do? Yeah, it'd smack her with the ruler and make her put it in the right hand. So then she came to the US and nobody cared anymore. And so she was able to switch. So if she had stayed in Portugal, though, as she had grown, as being continually conditioned to be forced to use the right hand, can those things change? Can we be socially cued to change those things? Yes. So some of it has to do with that as well, but definitely we have a preference, and not just for hand, but for leg and also for eye. When we're sighting or doing something like that, there is a dominant eye, dominant hand, dominant foot, and those things are genetically determined, but they can be socially influenced. All right. Excellent. And not that random, since we're talking about motor control. I think this was a great place to bring it up. No, that, stop it. Just want the line. All right, excellent. So those are the 3 regions and areas that we need to know for the frontal lobe. Questions on that? All right, so let's move to the parietal lobe. How many regions do we have to know in the parietal lobe?
2.
Excellent. Once again, as we have talked about, there is a raised ridge behind the central sulcus. What was that structure that is the raised ridge behind?
Postcentral gyrus.
Excellent. This postcentral gyrus is the location of our first area or region. And this first area or region is what is known as the primary somatosensory area. And with a name like that, what do you think its function is?
Sensory.
Excellent. So it receives sensory information, but what kind of sensory information? Soma. What does soma refer to again?
Body.
Body. So this is general sensory information of the body. And what part of the body do you think? Or what side of the body?
Contralateral.
There you go. And so over here on our post-central gyrus is our primary somatosensory cortex. And guess what? We have that exact same map that we were talking about before here on this side. Feeling from the contralateral side of our face, feelings from the contralateral hand, feelings from the contralateral arm, feelings from the contralateral trunk, and so on and so forth. Now, the trunk is the biggest part of your body. Do you think that has the most cortex then? No. The areas of cortex that control the different regions of our brain are actually, the amount of cortex is based on the amount that we use it. Notice here we have these two maps, these great pictures from your, this isn't actually from your textbook, but from another source. We see our primary motor cortex and our primary somatosensory cortex. Notice, as I mentioned, the largest areas, because if you think about it, if you remember, these are the areas that we had the most sensory receptors. Remember, we said our face and especially our lips, our hand, especially the thumb. Those are the areas where we have the biggest receptors. And then same thing for movement, large movements of the hands and large movements of the face that we're able to control, many control, right? How much emotion can you show with your trunk? Right?
None.
With your face, yeah, with your face, you can show you're angry, you're sad, you're aroused, you're sad, you're angry, you're happy, right? You're whatever, confused. You can do all of that emotion just by small facial changes, right? Show me what a confused trunk looks like, right? Yeah, I mean, again, we don't have that kind of motor control of it, right? And so again, we have these maps. These maps have a special name where the size represents the amount of the cortex, the amount of the brain that is involved with processing that information. And we call that a homunculus. And I think I have some pictures. There we go. Notice here, we actually have these great, it's not a great picture, but these great models that show the relative size of the brain that is involved in these processes. So notice for motor control, most of our motor control is in the hands and in the face, especially the lips, whereas very little in the legs and the arms. Notice can't really wiggle your ears, so really no motion there. Whereas again, our sense of feeling, hands and face, tongue and lips, right? Here we can hear, here we can see, right? Here we can smell. So we have, but again, the core of the body, not as much. So we have these funny looking guys that are the homunculi that are that representation of those two regions. Now, you may never thought of a homunculus before, but you're probably aware of this relationship between these two areas, because maybe you have that really old uncle who had a stroke. And we had a stroke basically as we saw, oh, actually, let's go back here. Notice there is this big, large blood vessel that basically goes down the central sulcus. And if this blood vessel gets blocked, then suddenly a portion of the brain is deprived of oxygen and they lose some abilities. And suddenly your uncle can't move his entire left arm and he can't feel anything on his hand, right? It's not like he can't move his arm and can't feel his face, but you get parallel deficiencies because both of these areas, now again, they're not identical. He may not be able to move his entire arm, but he may only lose feeling from the elbow down. But you get that similar contralateral effect when that tissue gets damaged from that lack of blood supply. Yeah, so exactly. So if this happened here, well, here, if this happened on the left side that we're seeing here, then it would be the right side of the body that would be the drooping as a result of that. All right. Questions on our primary somatosensory cortex. And again, because you asked the question, I want to make sure to emphasize this. On the exam now, if you notice for these two areas, there are basically 3 questions I could ask. I could point my arrow here, and I could ask 3 questions. Identify the structure, and what's the structure?
The primary motor cortex.
Nope. What is the structure?
Precentral gyrus.
There you go. Exactly. Absolutely. What is the area? Now you can say it, Ava.
Oh, primary motor cortex.
Excellent. What is the function of this area?
Voluntary control of contralateral skeletal muscle.
Perfect. Perfectly worded. Absolutely. So there you go. So most areas, it's just going to be identify the area, identify the function. But since these happen to be sitting on particular specific structures you're responsible for, there technically is a 3rd question we could ask there. All righty. Now, again, don't get confused because the brain has been turned around. But there is, oh, here, we'll look at this picture. So it's at least pointing the right direction. Notice on our frontal cortex, we had a frontal lobe, we had our prefrontal cortex, we had that speech area. And then notice back here in the posterior part of the parietal lobe. So let's draw this on our drawing. And it may slip around a little bit into the temporal lobe, but we'll emphasize it here. This is an area that is known as Wernicke's area. Why is it called Wernicke's area?
Bob Wernicke.
Oh, Bob Wernicke, exactly. This is also now known as general interpretive area.
So when you put the parentheses, are these basically interchangeable names that we can use? Okay.
Exactly. Again, as we know, many things were named by the anatomists who first planted their flag in it and described it, or physiologists who described it. And those are common terms that are commonly used, but they aren't necessarily useful terms because they don't tell you anything about it. Slowly, anatomy and physiology is getting better and better about this, where they're changing or adding a name that tells you more about what it does. The general interpretive area is another area that is lateralized. In right-handed people, like most of you, about 97% have Wernicke's area on the left side. And even left-handers like myself, about 75% have it on the left side. What Wernicke's area allows you to do is not to produce speech, but to make sense of speech and to make meaningful speech. Notice if someone has damage to Broca's area, if I have damage to Broca's area and you tell me to raise my right hand, I can do it. Now, this happens to be my left hand, but you get the right idea. I can follow directions. I understand speech, and I can understand speech. I just can't produce speech. People who have injuries to these 2 areas, that injury is what's called an aphasia. and it affects our ability to either produce speech or produce meaningful speech. And again, I think one of the easiest ways to see this is to see it in people who have injuries to this area. Like this one.
They're afraid, you know, boom.
What's this?
You know, they're big.
Can you guys hear that? Oops, I didn't mean to do
By
that.
advertising on YouTube, I catch the eye
Yeah,
of
yeah.
my ideal customer.
They're afraid, you know, boom.
Can you guys hear that?
Yeah.
Okay.
What's this?
You know, they're big. And right there, end of the football.
End of the football?
Is it influence the football right here? The cause that it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we have all of these, see?
I don't understand.
Well, it's pliable in the other, see? You don't understand it, but it applies in the long tree comes from there. And it comes source from the country out here.
Are you guys seeing this?
No.
No, we only hear it.
Okay, hold on. Let's try this again.
You know, the big and right there, end of the football.
End of the football?
It influence the football right here. The cause that it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we have all of these, see?
I don't understand.
Well, it's pliable in the other, see? You don't understand it, but it applies in the long tree comes from there. And it comes source from the country out here, see? All the under it, see, it comes up and finally come out here and goes out, see? Get it?
Sort of.
Yeah, it comes out here, see, it comes out here, here, and here, and it comes in, and it comes out, and bing. That's a good fly flow, see?
That's a good what?
That's a good flow goes fly, see, it flows, pull, or it gives, or kicks out, or bing, or whatever it is. We has no football fired out. It's just pulling, see, but they feel it. Oh, I guess you meant you have beds, see, I don't know.
Where do you live?
Where do you live? You live? You're up here, see?
Where do you live, Doc?
Where do you live?
Where do you live?
Where do you live is, well, if there's around here, around here. You?
You used to be a dentist.
Yes,
Where?
right here. The rest of the hearing, yes.
How many years were you a dentist?
How many? Holy Christmas. Oh, about 100 and, let's see, 130, about 40. No, gosh, what there is it? One plus 5. Wait a minute, no. It's a pretty close to get it this one day. It's about every year off, see. It's cross. Oh, that goes for about over 30, see, or 130 for down, see, in front of out for it's just straight against her, and then it comes back to the against her 4, and then goes out for the rest of the time. Third is not the servant, but the soldiers, where they have the soldiers of the Siberians and the suburbs and all the first Cerebral, and watch out in the short side, and they cross with the sugar 2 hours, 2 I heard low. Wow. God, these are terrible. Wow, these are terrible words.
These are terrible words.
They sure are.
Hard?
Yes.
What you're saying, you mean?
Yeah, that's what they do. They actually do them a lot harder. It's when they are in hard, go. And then when they go out, finally they quit, see?
Notice if you were at a Starbucks and these two were sitting at the table behind you and you weren't really paying attention, you might not notice anything wrong about their conversation. His speech has all the normal cadence and inflection of what you expect speech to be, right? But he's adding extra syllables, he's adding meaningless words or wrong words or confusion, and producing what is sometimes referred to as a word salad. And that's what happens with this condition. With this condition, he is able to, it impairs his ability to both produce meaningful speech and understand speech. You can see this in some of his responses. Notice she asks him, "Where do you live?" Now, turn that around. Ask me, "Where do you live?"
Where do you live?
Where do I live? Right? You ask me this and I turn the question around. I'm thinking about the answer. Do I really want to tell this person where I live? How much information, how much detail? And so I turn it around. You ask me, "Where do you live?" And I turn it around. Where do I live? But she asks him, "Where do you live?" And he repeats it back exactly to her. Where do I live? Or where do you live? You know, she says, "Where do you live?" He says, "Where do you live?" He repeats it. He's trying to make sense of the words. He doesn't understand what the words are, and he doesn't come up with a meaningful question. But when she asks him about being a dentist, clearly he remembers about being the dentist. He even makes the joke about in his mouth. But then she asks him about how long. And instead of just being able to come up with a number, he comes off with this big all explanation because he can't come up with a number. And I've watched this many times. And I believe what happened is he went to a dental school, became a dentist, and then went to war for a few years. And then when he came back, was a dentist for the rest of the time, because he was talking about the soldiers and around the short side and driving the tanks and all of that kind of stuff. So clearly he is able to somewhat understand some of the things that she's asking, and he can produce some meaningful responses. But how much he fully understands isn't fully clear. And he's also, right, he's pretty good natured about this whole thing. He makes the joke about the teeth when he's talking about the dentist, but at the end there you see a little of his frustration. These are hard words, right? I try to formulate them in here, and then when they come out, they just quit. So clearly he is aware of that he's got some impairment, but it's hard to know how much he's aware of the impairment that he is, because it's very hard to communicate with him. So this is an example of someone who has that Wernicke's aphasia. Notice he's capable of producing speech in cadence, and it sounds just like normal talk, but it isn't necessarily meaningful words.
That's so sad.
Yeah, I'm sure he's dead by now, though, so it's okay.
Okay.
All right. Augustina, great question. The answer is, oh, yes. So yes, Sarah, to answer your question, yes. Things like Wernicke's or Broca's aphasias, these are also things that can be caused by a stroke. Now, Wernicke's area is the ability to interpret and to make speech, right? For some people, they're still able to read and write perfectly, but some people aren't able to read and write. But it's not hearing. Hearing gets us ahead of ourselves a little bit, but as we'll learn, our auditory system, our hearing, is down here in the lateral lobe. So in our lateral lobe is where we'll find our, where we'll find the area that where if this part's damaged, this is where one of the ways you can be deaf and not be able to perceive sound. Now, we have one more big issue with this lateralization of Wernicke's area. Then what the heck is on the other side? And once again, this is something that is really, really interesting. On the opposite side, oops, hold on. Well, we'll go ahead and erase that anyways. We'll deal with that later. on the opposite side is spatial awareness. Again, the way we know this is by people who have had injuries to this area. Someone who has damage to basically what would be Wernicke's area on the right side, you show them a picture of a potted plant and you ask them to draw it. And what they will draw is that. They completely ignore the contralateral side of things. And I mean the entire contralateral side. I had a student, God, I guess with COVID has kind of felt like it stopped time. So it seems weird this way, but so it must have been 6, 8 years ago now. Like 6 years ago, I had a student whose boyfriend had been in a car accident and had a traumatic brain injury to his spatial awareness side. And he would get angry in the shower about that person who was standing too close to him because his left arm would be in the way. He would only wash the right side of his body. He would forget to dress the left side of his body when he was getting ready in the morning. So she would have to help him to remember to do this. It wasn't a complete deterioration. So sometimes he was aware of it, but a lot of the times he would completely ignore the left side of the world and then even the left side of his own body.
So if he was looking in a mirror, would he only see like half of himself?
Yep.
That's weird.
The brain is cool.
Oh, that's so weird.
Absolutely.
Okay, so for Wernicke's area, it's lateralized to the left side only.
Again, in right-handed people, 97% have it on the left side. In left-handed people, it's even 75. So yes, most people have it on the left side.
And the function is to
interpreting speech and producing meaningful speech.
Okay. Thank you.
All right. Excellent. All right. I'd hoped to be able to finish the last 2 lobes, but I don't want to rush it. So we'll go ahead and stop here for now, and then we'll pick up with our occipital lobe and temporal lobe in the next class. As you can see, it won't take us long, but I still want to have a meaningful conversation, and then we have to move forward from that. So we've got halfway through our telencephalon. We'll finish the rest of it off when we get back on, I guess, Monday. Yeah, on Monday. So on Monday, we will do that. In fact, I think on Monday, we'll start with the nervous tissue. We'll start with the brain anatomy because we're more behind on this brain anatomy stuff, and then we'll get back into lecture after. So I'll figure it out and we'll do what we can to start catching up. All right. Any questions on any of this? All right. Then that is all we have for today. You guys study hard this weekend. Like I said, I'm glad you guys think the brain is interesting. Again, being a neuroscientist, I'm biased, but I think it's really cool as well. So hopefully, like I said, while it's not necessarily the most intuitive information, hopefully we can make it interesting enough where it will make sense to you.
Are our grades and tests going to be released today or no?
Thank you. Yes, the grades are actually posted. Absolutely, the grades are posted. I will release the grades right now. In fact,